Good morning, good afternoon, good evening, wherever you are in the world, and welcome to this scientific symposium. It's marked the end of these celebrations of 2021 edition of the World Blood Donor Day. It's my great pleasure to welcome all of you. Yesterday, we had a lot of personalities from the Minister of Health and to people from the W Health Organization, the International Federation of Red Cross, and the International Society of Blood Transfusion and Donor Organization, taking the floor to welcome all the people and to thank all the donors that gave their tribute to save life throughout the world. And in evening, we had the big concert, we joined in the big concert, and so this is the scientific symposium ending this day, and I would like to welcome all of you and to listen to all the scientific relevant personalities that today will take the floor and will give us important insight into the field of blood transfusion and into the field of the future of blood transfusion, also in conditions as this pandemic was, in which really the availability of blood and blood products to all the people can be threatened. So let's start with the introduction of the first session. Well, it is my pleasure now to welcome the first of our speakers, Dr. Lorenzo Montrasio, who currently leads the work on equity of access of innovative treatments at the Human Rights Directorate of the Council of Europe. And before joining the Council of Europe, Lorenzo served for more than 15 years at the Italian Medicine Agency.
Okay.
Where he was responsible for the marketing authorization of biological medicines, including vaccines and plasma and blood-derived products. And in the blood sector, Dr. Montrasio coordinated the European Pharmacopoeia Cooperation Program with blood establishment and testing laboratories in 26 countries from 2018 to 2020. And since 2017, Lorenzo volunteers for a development program on rural healthcare at the St. Luke Hospital in Cameroon. So please, Lorenzo, take the floor for illustrating us the Convention of Human Rights and Biomedicine, the Oviedo Convention. Thank you, Lorenzo.
Dear Vincenzo, dear colleagues, thank you very much for this very nice introduction. With my presentation, I will shortly introduce the Oviedo Convention. That is an international convention on human rights and the application with respect to the application of biology and medicines. I just hope that you can see my slides because there was some problem of connection. Can you just confirm, please? Hello?
No, we are seeing you, but not
your slides.
Seeing your slide.
Not my slide, so sorry about that. Just a second. So it's always...
we saw before.
There is a problem. It's always on the wrong moment. Can you see now?
Yes.
Okay,
yes,
that's great.
Now we can see.
Okay, so I apologize for that. Okay, so as I mentioned, I will briefly introduce the Oviedo Convention. That is actually the Council of Europe Convention on Human Rights and the application of medicine and biology. Just a few words regarding the Council of Europe. Council of Europe is an international organization based in Strasbourg and established after the Second World War in 1949. This organization brings together 47 member states that are party to the European Convention on Human Rights, whose application is monitored by the Court of the Human Rights based in Strasbourg. The role of this organization is mainly strengthening human rights. And the means that are provided to this organization by the treaty is the activity of standard setting, monitoring, and cooperation between the parties. This makes a clear distinction between the Council of Europe and the European Union. But the countries that are part of the Council of Europe are the same countries, those countries that are part of the European Union, and plus we have a party of the Convention, Russia, Turkey, and the country of the Caucasian and Balkanic areas. Going to the Oviedo Convention. The Oviedo Convention provides a common framework for protecting human rights and human dignity in both long-standing and developing areas concerning the application of biology and medicine. And it was recognized by the drafter of the convention that the biomedical fields actually is a source of great benefit for human beings, and in particular for human health. But at the same time, this development and this benefit could bring the possibility to intervene on and control human life. And this raised the concern that could be summarized in these 3 points that are the points that the Oviedo Convention is focused. And in particular, this concern could regard the practices that could be contrary to human dignity, such as the eugenic practices, or area where autonomy should be protected, patient autonomy, or vulnerable persons should be protected in both research activity and the medical practice. And another area of concern that was recognized at that time was the risk of commercialization of human body and its part, including activities related to the trafficking. The Oviedo Convention, with these perspectives, was a pioneering work, and still now is the only binding legal international instrument to provide human rights in the healthcare sector. It's a living instrument, and the drafter provided the fundamental principles and at the same time mechanisms for re-examination. The convention, I dare to say, that proved to be relevant during this COVID-19 crisis. And we have clearly seen that the Oviedo Convention remains a relevant reference also for the European Court of Human Rights, faced with an increasing number of cases related to health, and in particular to the protection of privacy, to access to treatment and health care, and in general, the protection of and against the risk of discrimination. The parties to the Oviedo Convention, this is just to show where we are. This convention is in a certain way a parallel convention to the convention, the European Convention on Human Rights, and is under the same umbrella. So we have under the Convention of the European, the Protection of Human Rights, we have 47 countries. The countries that decided to ratify the Oviedo Convention are for the moment 29. 35 signed the convention. This convention was open for signature in 1997, and the process, I dare to say, that is still ongoing, and that there is an increasing interest to join and complete in the process for the ratification of the Oviedo Convention. The impact of the Oviedo Convention is clearly more relevant for countries that have ratified, but this is not the only countries where this the Oviedo Convention made a difference, and several of the principles that are included in the Oviedo Convention have anyway included in several legislation in Europe and around the world. And even the European Union has taken the Oviedo Convention as a reference for several directives and also for the European Charter of the Fundamental Rights. And impact also is relevant at the global level. But when it comes to the principle, the core of this Oviedo Convention, I would like to mention, first of all, the Article 2 that express the primacy of human being over the sole interests of the society and science. And this is really the inspiring article. And in this light should be read all the Oviedo Convention. And it brings the fact that when there is a conflict between the human being and the interest of the society and science, human beings should prevail. And then a second important principle is the equity of access to healthcare. And this provides obligations to country to provide healthcare services with appropriate quality, taking in consideration the available resources. And in the Council of Europe, there are countries with a huge difference in healthcare resources available. The effort that the convention asks to governments and the party is to make any effort as much as possible to provide quality healthcare. The principle of protection of private life and the right information brings directly to the Article 8 of the European Convention of Human Rights, and this becomes extremely relevant when it comes to the judgment of the European Court of Human Rights. And this deals with the right to have for the personal information of health, and the right for privacy, and the right for patients and citizens to know which information are collected regarding his or her health. The principle of consent. The principle of consent affirm that no one may be in principle be forced to undergo an intervention without his or her consent. And the consent should be free and informed and given on the basis on objective information. There are specific articles in the Oviedo Conventions that also deal with the specific situation where this right can be limited. And this include emergency situation and situation concerning the protection of person who have a mental disorder or the protection of person not able to consent. Last but not least, the principle of prohibition of financial gain and disposal of part of human body is Article 21 and brings us directly to the core of this presentation. That is actually the Article 21 that stated that the human body and its part shall not as such give rise to financial gain. This is an important principle that has been taken in consideration in several regulations regarding the blood and the blood products. These include the EDQM guided preparation, use, and quality assurance of blood components, but also the Convention Against Human Organ Trafficking, as well as the European Charter, as mentioned before, and the directives on blood, cell, and tissues, and WHO guidelines. And this principle of prohibition of financial gains, for this principle, it's important that there are clarification. And that's why the Committee on Bioethics, together with the Committee of the EDQM on transplantation and blood transfusion, approved and adopted a guideline that give a support in the implementation, while clarifying some concepts that are particularly important with this regard and with regard to no financial gain principle. The first idea is that during a donation and related to the donation, there is a financial neutrality. And this means that the reimbursement, compensation are perfectly fair, but this should be transparent and justificable based on the expenses or the loss of earning. So the idea is that the financial aspect should not become in any situation the main motivation and the strong motivation for giving blood. A second aspect that is explained in this guideline is that even when there are medical and technical services, such as the transformation of blood and plasma in medicinal products, it's absolutely in line with this principle to have a fair remuneration. But this, again, should be based on the principle of transparency, transparency of cost, transparency of prices, and again, because we have to consider that when we start with a starting material, a raw material like blood, that is given for free, there should be a fair approach also in the remuneration for these approaches. And these guidelines also clarify another aspect, is that it's absolutely compatible with this principle of no financial gain, and it's mandatory also according to the Oviedo Convention, the compensation in case of undue damage resulting from donation. Having said that, and just to go briefly to the conclusion, the take-home message are that the Oviedo Convention is an international instrument, providing a legal framework for protecting human rights in the field of biomedicine. And relevant in the field of blood donation is the principle that the human body and its part shall not raise to financial gain. And this is in line with the idea that there should be no commodification of human body and its part, and there is a need to protect against exploitation. Because it was also recognized that financial gain may have an effect of influencing the most vulnerable people in society and expose them to exploitative actions. And finally, what I would like to raise is that this principle and the Oviedo Convention really contribute to promote the altruistic donation and promote and increase safety and quality of donated human parts. And this is particularly relevant when there is uncertainty in the epidemiological situation of potential contaminants, and when there are no specific testing methods. And finally, my key message is that this principle really contributes in maintaining a donation system in which people can trust. Having said that, I would like to thank you for your attention, and I would like to introduce the next speaker, that is Vincenzo De Angelis, that everybody knows, and will explain to us the Italian blood system and as an ethical and effective network. Thank you very much for your attention. Please, Vincenzo, the floor is yours.
Thank you very much, Lorenzo, for your really nice presentation, which is the basis of our job and of our organization. So it is my pleasure to explain, to say something about the ethics and the efficacy of the Italian transfusion system. May I have my slide, please? And, as you mentioned before, the basis of our job is represented, of course, by the ethics of our organization. That, however, must be funded in some way. And as Benjamin Franklin said, in this world nothing is certain except death and taxes. And nobody likes to pay taxes, and also Italians do not. But taxes are essential to fund the many things that are necessary to grant the fundamental rights to the people, as the right to health. And universality, equity, solidarity, and high-quality health care for all citizens are the values and the strengths of the Italian National Health Service. And it provides high-quality health care to all citizens for hospital care, emergency care, primary care, without discrimination based on income, gender, and age. The next, please. But a well-performing health national service requires a transfusion system based on ethical principles and social values, fully compliant with European legislation. And in Italy, voluntary, periodic, responsible, anonymous, and non-remunerated donation of blood is the basis of the system, and it is the core value of donor association responsible for the recruitment and retention of donors. And the transfusion process is unitary, and blood establishments are exclusively public. And any charge of money to patients for blood transfusion is forbidden by law. The next, please. And health authorities and regulatory bodies, European, national, regional, blood establishment, scientific society of transfusion medicine, donors associations, universities, pharma industries represent altogether the many different actors and stakeholders of this system. The next, please. But Italian blood system strategic goals are defined by the law, and they encompass self-sufficiency of blood and blood products, including plasma-derived medicinal products, promotion and continuous development of voluntary non-remunerated blood donation, high quality and safety of blood products in compliance with European regulatory provisions, appropriate management and clinical utilization of blood resources, and continuous development of transfusion medicine aligned with scientific progress. The next, please. And by this reason, every year, the Minister of Health defines the National Self-Sufficiency Program, which identifies the production levels required, the resources, the criteria for funding the system, and for ensuring the exchange of blood and blood products among regions with the aim of a national self-sufficiency. The next, please. But it is worth mentioning that a full integration of the blood system of donors, volunteer association exists in Italy. The associations are funded by the government in order to perform not only campaigns for blood donation, but also donor recruiting and retention, schedule for donations, and in some regions also collection of blood together with the blood establishment. The next, please. And in 2020, the year of pandemic, the system took care of more than 1,600,000 donors, out of which only 33% are women, and extremely worrying, the majority of donors are in the age between 35 and 60. Gender disparity and young involvement are a matter of reflection and must be emphasized in this 2021 World Blood Donor Day edition. The next, please. But these enthusiastic and motivated donors gave in 2020 almost 3 million donations, leading to the transfusion of more than 2 million units of red blood cell, more than 200,000 units of platelets, and roughly the same amount of units of phosphorus and plasma, which means that 1,653 patients are transfused per day in Italy, one transfusion every 9 seconds, and one transfusion of plasma-derived medicinal product from national plasma every 10 seconds. We would here to say thank you to those splendid persons. The next, please. As a whole, Italy is self-sufficient, and the number of whole blood units collected is always higher than red blood cells transfused in the country. The next, please. However, this is the net result of regions where collection of blood is both efficient and effective, and from those regions, a regular supply to regions where the need is higher than the local donations occurs. Differences in availability of blood donation may only in part explain these discrepancies. We need to take into account also differences in health services, complexity among regions, with patients moving to national reference tertiary care hospitals present in few regions in Italy. The next, please. But also epidemiological data, peculiar of some regions, may be as well determinants of discrepancy among need and supply. I would just here to mention the fact that the two major islands, Sardinia and Sicily, account for roughly half of thalassemia patients in the country. The next. And to favor and help the availability on time of blood where it is needed, an electronic support is active at the National Blood Center and visible on the website, showing every day where and how much blood is available and where and how much is needed. The next, please. And a special and distinct national program is endorsed by the legislation to promote self-sufficiency of plasma-derived medicinal products in order to provide patients in a systematic and sustainable way with a prompt and continuous availability of a defined set of plasma-derived medicinal products with the highest level of quality and safety. The next, please. In Italy, toll manufacturing is by law the only possibility to fractionate into medicinal products plasma collected and tested by the blood establishment from voluntary non-remunerated donors. There is not a unique national tender, but different regions join in partnerships needed to reach adequate volumes of plasma to be fractionated. And in the last years, tenders have been finalized by 4 distinct interregional agreements and awarded to 4 pharmaceutical companies to fractionate Italian plasma. And this led to a basket of many plasma-derived medicinal products, whose availability is expected to increase the satisfaction of internal need with products coming from national plasma. The next, please. And with more than 843,000 kilograms of plasma collected in 2020, Italy is the 3rd European country for volume of plasma fractionated. And the fractionation rate of 14 kilograms per 1,000 inhabitants is what we consider a good success. However, there is a pronounced difference in the country between regions collecting plasma for fractionation at a rate up to 33, 34 kilograms, the next please, inhabitants, and other collecting less than 6 kilograms per 1,000 population. And many interventions are in progress for the improvement of plasma collection on national basis. The next please. And also, in the case of plasma-derived medicinal products, trading and offsets among regions defined and harmonized by the legislation allow the availability of different products nationwide. The next. Thus is the basis of the important contribution given by the fractionation of Italian plasma to self-sufficiency in medicinal products, reaching the remarkable figure of more than 70% for albumin and IVIG, and 80 to 90% for antithrombin-3 and prothrombin complex concentrates. We need to do better, because this result is, of course, even more important and strategic in conditions as represented by this pandemic, where availability of source plasma from abroad is decreased, thus leading to possible shortage in supply of medicinal products essential for patient survival. The next. And finally, promotion and implementation of collaboration agreements for the export for humanitarian purposes is the matter of a ministerial decree dealing with all the medicine from national plasma that are produced in excess of national need, as for plasma-derived factor 8 and factor 9. And in these years, more than 75 million units of plasma-derived factor 8 and about 4 million factor 9 fractionated from Italian plasma has been gifted to different countries where people with hemophilia are receiving inadequate or no therapy due to reduced availability or even unavailability of coagulation factors. The next. And in conclusion, in Italy, we are continuously working for improvement of our blood transfusion system. We are proud of it. It promotes the safe recruitment and retention of voluntary non-remunerated blood donors. All health is protected by the system. And they are the irreplaceable basis for the self-sufficiency of blood and blood products. It ensures blood availability free of charge to patients in all the country, thanks to an exchange of blood and blood products among regions, promoted and supervised by the National Blood Center. And it also offers a concrete humanitarian aid through the export of plasma-derived products. But it is also strategic in ensuring the independence or near independence for different essential plasma-derived medicinal products with a relevant money-saving effect. The last one. And with this, I would like to thank you, all of you, for kind listening and give blood and keep the world beating. Thank you. And having said that, we are going to the first presentation of this introduction. And the presentation is by Dr. Yun Mariuningsi, Medical Director with specialization in transfusion medicine and a doctorate in biomedical sciences. At present, she is the team leader for blood and other products of human origin, technical standards and specification unit at the Health Products Policy and Standard Department of the WHO headquarters. She has a wide and appreciated experience in the field of transfusion medicine, gained through different activities: Blood Services Program Coordinator at the Ministry of Health of Indonesia, Chairman of Indonesian National Blood Services Committees, and many other activities. She is also at present the chairperson of the Indonesian Association of Transfusion Medicine and served as a regional director at the International Society of Blood Transfusion for Southeast Asia region until 2019. The floor is yours.
Thank you, Dr. De Angelis. Good morning, good afternoon, good evening, everyone. I'm very pleased to be part of the scientific symposium in related to 2021 World Blood Donor Day celebration, hosted by the Italian National Blood Center. And thank you for asking WHO to share the action framework to advance universal access to safe, effective, and quality assured blood products 2020 to 2023. Let me share my slide. Yeah, I hope you can see my slide from the screen. So in this presentation, I will briefly explain the challenges and reasons why this action framework is needed, its aims, strategic objectives, as well as the outcomes. We are all aware that blood and blood components are critical for treatment of bleeding, for example, due to birth delivery, severe injuries, and surgical procedures, and also to manage the life-threatening condition, for example, for hemophilia, thalassemia, immune deficiency, and cancer. Plasma-derived medicinal products, which is part of blood products such as albumin, blood coagulation factors, immunoglobulin, and other factors are also important for managing certain medical conditions. Based on the Global Database on Blood Safety 2015, we identified some challenges in blood services. So the first challenge is inadequacy in policy, regulation, governance, and financing, in which the data shows that only 60 to 70% member states have blood policy legislation and oversight system. The second challenge is insufficient supply of blood products, in which the data shows that there are still 66 member states with blood donation rate less than 10 per 1,000 population. The third challenge is deficiencies in safety, effectiveness, and quality, where only 80% of donated blood was tested in low to medium income countries. The first challenge we identified is lack of availability of plasma-derived medicinal products. This is because of limited use of blood components that lead to low volume of plasma available for fractionations. The fifth challenge is suboptimal clinical practices, because clinical use of blood, hemovigilance system, and patient blood management are not implemented. And the last challenge we identified is poor access to blood during emergency situation, because no blood supply preparedness system. WHO has responded to these challenges through issuing several WHO resolutions. The one in 2005 designated 14 June as a World Blood Donor Day, and the last resolution in 2010 emphasized the importance of availability, safety, and quality of blood. And to urge government to put more attention on blood services, blood products has been included in WHO list of essential medicine. And furthermore, the 2018 International Conference on Drug Regulatory Authorities called for strengthening hemovigilance system to monitor the safety of blood donations and transfusion. Countries that implement WHO resolutions and guidance documents on blood, mostly, of course, developed countries, are making progress in providing safe blood and blood products. However, this progress has been slow in many countries. Therefore, there is a need to push for implementation of WHO guidance at country level, particularly in low to medium income countries. The action framework aims at strategic direction to global efforts to address present barrier to safe blood, and it also responds to the WHO Resolution 2010 on availability, safety, and quality of blood products. The action framework also aligns with WHO General Program work and strategic plan for regulatory support activities for health products. And this document consists of implementation of resolutions, goals, and strategy. And finally, this action framework also can be used to build the partnership and fundraising. 6 strategic objectives have been set up as proposed action to cope with challenges. So one is the appropriately structured, well-coordinated, and sustainably resourced national blood system. Second is appropriate national framework of regulatory controls, functioning and efficiently managed blood services, effective implementation of patient blood management, effective surveillance, hemovigilance and pharmacovigilance, and partnership, collaboration, and information exchange. Its strategic objective has some outcomes. So the outcome of the first strategic objective are that national blood system is integrated into the national health system. National blood system is adequately and sustainably costed, financed, and budgeted. National policies and decisions are made through good policy process and risk-based decision making. And the last outcome for this strategic objective is adequate and safe blood supply during emergency situations. The outcome for strategic objective 2, that the national blood regulatory system is in place in maturity level 3, means that the blood regulatory system is stable, well-functioned, and also integrated into the health regulatory system. And another outcomes is regulatory mechanisms are in place to oversight blood substance and IVDs. Quality assessment is carried out for blood substance and IVDs, and performance of blood substance and IVD is assured through use of international standard. The outcomes for strategic objective number 3 are achievement of 100% voluntary non-remunerated donation, protection of donor, and promotion of repeat donation. A functioning quality system is in place, blood services are efficiently and cost-effectively managed, and availability of volume and quality of plasma for PDMPs is increased. There are 2 outcomes for strategic objective number 4, which are good patient blood management is practiced based on national clinical guidelines and practice standards, and a quality system is in place in hospital for all pre-transfusion testing. For strategic objective number 5, there are 3 outcomes. A national system for standardized data collection, reporting, and mechanism is in place. Hemovigilance and pharmacovigilance are also in place, and WHO Global Database on Blood Safety provides comprehensive and accurate data on global status of blood availability, safety, and quality. And finally, for the strategic objective number 6, we set up 3 outcomes, which are training programs on key functions of national blood system are capacity for external assessment and accreditation of blood establishment is available. Capacity to evaluate relevant new technologies and other innovations are incorporated to the national blood system. And the last outcome for the strategic objective is regulatory capacity is strengthened through collaborative capability building. So since last year, we have conducted some activities to implement this action framework. So 2 guidance has been developed in related to strategic objective number 3. So these guidance are guidance on centralization of blood donation testing and processing, and guidance on increasing supply of plasma-derived medicinal products in low to medium income countries through fractionation of domestic plasma. These two guidance has already been published in March 2021. So why we put these two guidance as a priority to be developed? Because we believe by centralizing the blood donation and processing, the member state can standardize the quality of blood and also can increase the volume of quality plasma for fractionation. And this year, we hope we can develop and publish another 3 documents, which are education module on update clinical use of blood and policy brief for the implementation of patient blood management in related to strategic objective number 4. And the other document is education training modules on hemovigilance in related to strategic objective number 5. And another activity is technical assistance in the forms to conduct the workshop, training program, country assistance, and training program. So this activity, of course, should be conducted at all 3 WHO level, global, regional, and national, and in the 6 WHO regions. So thank you for your attention.
So thank you very much. Very interesting presentation. We have few minutes saved during this session. So I think that if someone put on the chat one question, we have time for one question. Yeah, I have one question. Is how the health of voluntary non-remunerated blood donors is protected by the system in Italy is for us. And is Andre from the WHO regional office in Africa. The answer is quite easy. It is protected by law by a set of provisions starting from the doctors of the blood transfusion services taking care of the donor health protection, and by also a set of blood tests that must be done during blood donation at least every year in order to be sure that the health of donor is protected and we are not doing any kind of harm to the donor. And so it is a sort of donor protection mediated by the fact that the blood transfusion system are in charge also to be sure that no harm is done to the donor, and all those provisions are set up by the national legislation. I have no other questions in the chat, so I would like to just to have a question for Dr. Mariuninzi. So I'm really very, very happy for that guidelines on increasing the collection of plasma for the production of plasma-derived medicinal products from domestic plasma. So it is, in my opinion, very strategic. The fact is that domestic plasma must reach a volume, a defined volume, in order to be attractive to the pharmaceutical companies to be fractionated in the medicinal products. So do you think that if this volume is not reached because of the small volume of plasma collected in a nation or the small size of the nation, we also could imagine some kind of, let me say, agreements among nations in the regions in order to reach adequate volume of plasma for fractionation?
Thank you, Dr. for the question. So fortunately, I came also from the developing country, so I can share my experience. So we indeed have a difficulty to achieve the adequate volume of quality plasma for fractionation. So the first problem is because some clinicians still like to use the whole blood for transfusion. So again, we don't have enough volume of plasma for fractionation. And I think also because the quality of plasma cannot fulfill the requirement for the fractionation. So that is why WHO last year developed two documents, which is actually complement each other. So because by centralizing the blood testing and processing, then we think that this is one of the mechanism actually to increase the volume of plasma and also to standardize the quality of plasma. So then if there is a sufficient volume of quality plasma, then the developing country can move to the fractionation. And talking about the fractionation in the document, we also mentioned that maybe the first step for the developing country is to do the contract fractionation. And I think your idea also to gather all the plasma from the same region is maybe can become one of mechanism for the developing country to be able to fractionate their plasma. Thank you.
Thank you to you. The last question coming from Michele Lozano, who asked if I think, but it's a question I think that it is to everybody in the audience, if Europe in general and Italy in particular will succeed in ensuring self-sufficiency in plasma-derived medicinal products from voluntary non-remunerated plasma donation. Well, I hope, of course, and we all think that this is the way we are straight on. It is for sure that you have to complement the self-sufficiency with products coming from the market because it is clear that the intellectual property of some products coming from fractionation are not always and freely available. So when we discuss about the self-sufficiency, we discuss about the capability of a country or a region, as you said, to be, let me say, not really strictly dependent from the market, but having the capability to access the production of a good amount of products, and mainly the strategic ones, the drivers ones, as albumin, immunoglobulins, and so on. So this is something that we hope we need, and we have to go on in this way. Well, I think I don't see any other question on the chat, so I think that we can go on with the introduction to the first section of the symposium, which is dedicated to the international perspective and initiative in horizon scanning. So let's open the first session. And it is really my great pleasure to welcome and to give the floor to the chairperson of this session, that are two really good friends. And so it is my pleasure to introduce Dr. Cynthia Osman and Dr. Nicola Magrini. Dr. Cynthia Osman is a medical doctor, specialist in hematology and transfusion medicine. She has a Master of Science degree in clinical epidemiology. She served present as staff member at St. Queen Blood Bank in Leiden, involved in the department of transfusion medicine with specific expertise in patient blood management, as clinical consultant of transfusion medicine, and also as hematologist at Erasmus University Medical Center. Among the many activities of Dr. Cynthia Osman, I would recall that she has been, until a few days ago, the chair of the International Society of Blood Transfusion Working Party of Clinical Transfusion. And she was a really fantastic chairperson, and I would like to thank Cynthia for all the job that she did in this activity. And Dr. Nicola Magrini is at present the General Director of the Italian Medicine Agency, so it is the regulator in Italy, but until 2020 was Secretary of the World Health Organization Essential Medicine List, and in the Essential Medicine List, of course, plasma-derived medicinal products are present. So he is well informed on the topic that we are discussing. And not willing to enter the really very large CV of Dr. Magrini, I just would like to recall that in the last 5 years has been member of the WHO Ethics and Research Committee, of the WHO Ebola New Treatments Working Group, member of the GARDP DNDi Scientific Committee for the Discovery of New Antibiotics. He published more than 80 papers in international peer-reviewed journals, 2 books in the field of pharmacology, and served in many different groups of the World Health Organization drafting on working of different guidelines in pharmacology in the use and appropriate use of medicines. So I give the floor to the chairpersons and wait for them to lead this first session on international perspective and initiatives on horizon scanning. Please.
Well, thank you very much, Vincenzo, for these kind words. It's a great honor and, of course, great pleasure to chair the session today together with my co-chair, Dr. Nicola Magrini. And let me share my screen first. So can you see my screen? So this session is about convalescent plasma and hyperimmune immunoglobulins and lessons learned during the COVID-19 outbreak. We have a great set of speakers from all different societies and organizations, from WHO, from the Council of Europe, from the European Commission, ISPT, and the European Blood Alliance. So just a few slides on where do we stand now. What is the role of the passive immunotherapy? So meaning COVID-19 convalescent plasma and hyperimmune immunoglobulins. So at the start of the pandemic, there were a lot of questions that needed to be answered, and still a lot of questions are unanswered. So when should we administer this in the course of the disease, and who should we give it to? What about the immune compromised patients? What about the product characteristics, the titers on the neutralizing antibodies in the product? So there was a group of dedicated researchers from the Cochrane, from the UK, from Australia, from Germany, and the Netherlands, who performed a living systematic reviews during the pandemic, meaning that every time new evidence become available, then they updated their systematic review. So this is already the fourth living systematic review, looking for high-level evidence on the role of convalescent plasma and hyperimmune immunoglobulin. So at the moment, 13 studies have been, randomized studies, controlled studies have been found with almost 50,000 participants that investigated convalescent plasma. And it should be said that all but one study included participants who had moderate to severe COVID-19 disease. So the studies were mainly in the hospitals and in countries all over the world. And they had the following key messages that up to now, the Cochrane group was very confident that convalescent plasma has no benefits for the treatment of people with moderate to severe COVID-19, but still it is very uncertain about the effects of convalescent plasma for people with mild or even no symptoms. And what about the patients who are immunocompromised, who do not have antibodies against COVID-19? So still, this living systematic review is ongoing because there are still 130 ongoing, unpublished or recently published studies. So we're still awaiting for new evidence coming in. And this is just an example on one of the first papers that was published on 17 immunocompromised patients, in which almost all but one experienced an improvement of the clinical symptoms after receiving convalescent plasma. So it might be plausible that for this type of patients, convalescent plasma is still very valid. So what about hyperimmune immunoglobulins and the effect on it? Well, this is also very important, and several initiatives have been started to produce hyperimmune immunoglobulins as a therapy. But still new evidence is needed to come in regarding RCTs. And this is just one of the RCTs that are ongoing here in the Netherlands on 86 patients who are randomized between convalescent plasma hyperimmune immunoglobulins with a control group, just normal immunoglobulins. So it's very interesting to know the results of this and other studies as well. And we're happy if this study can finalize somewhere this year. So I would like to conclude that one big lesson while collaboration is really vital to help each other as much as we can, to share resources and scientific data, and to learn from each other to make the world safe again. So now I will hand over to my co-chair, Nicole Magrini. Thank you.
Thank you, Cinzia. Thank you all. Thank you, Vincenzo, for these very kind invitations and probably 2 nice words for my introduction. I'm very glad, however, to introduce my former colleague, Yuyun Marioning-Si, who leads the blood and other products of human origin at WHO in Geneva, to give a speech on WHO recommendation on collection and use of COVID-19 convalescent plasma. And let me just give an historical hint to share with the many attenders of this important day that in 2013, WHO, after a very crispy and interesting editorial on the New England Journal of Medicine by Harvey Klein, introduced and reclassified all blood products in the essential medicine list, and also introduced the blood as such and red blood cells as an essential medicine. So please, Yuyun, you have the floor.
Sorry, Nicola. We have just for problem because a slight modification of the program. You had the oldest one, not the last one. So in fact, the first floor is to Dr. Michele Lozano, and then to Stefan de Spiegel, and then to Yuyun Marioning-Si. I'm sorry, so sorry, because
sorry indeed.
We had this modification 2 days ago, and probably. So we had just sorry for that, and sorry to Yuyun. So Michele, please.
Thank you very much, Vincenzo, and thank you. Sorry, Dr. Magrini, for having to change the program. And let me start sharing my slide. Rising to such a small beta coronavirus has been able to produce such a big outbreak and such a big.
Sorry, we have some problem from your audio. Maybe the connection of your jacks and the computer. Please try again.
I
okay.
am checking.
Thank you. It's great. Can you share again, please?
Okay. Can you see now my? Oh, okay.
No. You need to share again. Okay, now it's working. Thank you.
You're welcome. Okay. This is the beta coronavirus that I was mentioning, and to describe that is really, for me, is really surprising that such a just a respiratory virus has been able to produce such an outbreak around the world, such a pandemic. And obviously, this pandemic was truly a surprise, and that found us without tools to treat the disease. That's why when that in the early days of the pandemic, we tried to use different therapeutic approaches, and from antiviral drugs to even to using anti-malarial drugs, and obviously we tried what we thought that could be a good approach, which was to administer convalescent plasma to patients, to severe patients. I will follow this very interesting paper published by the ISBT Convalescent Plasma Working Party just a couple of months ago in Vox Sanguinis, where the group of authors just look at the difference between high and low middle income countries. And in that analysis, I found that actually there are challenges that are common not only to high but also to low middle income countries. And those challenges can be divided into the different aspects of the plasma collection. One is how to identify the donors, because obviously you would expect that to identify people recovering from the infection. So there are different strategies to identify such a donors. One could be the self-identification, but also actually what we did in our case, which was going to the hospital database and to find people discharged after suffering the disease. And also you could try to identify such a donors from mining in different electronic medical records and registries. And the second point is try to recruit the donors. So you need to access the contact details of the potential donors. But obviously, in the conditions of the early days of the pandemic, where with the lockdown in place in many places of the world, it was really difficult to access eligible individuals. And obviously, at that time, it was very important for the donors to assure the safety of the selection and the donation process. Once you have identified the donor, then the next step is the establishing eligibility. And to do that, obviously, you needed to have evidence of COVID-19 infection. And one source, obviously, was the medical history of the potential donor. So as I mentioned, you need that the donor had recovered from the infection. And so a lot of people waited between 14 to 28 days after the recovery of the infection to give the plasma. And some of the countries, some of the centers also required a negative nasopharyngeal swab molecular test for SARS-CoV-2 in order to accept the donor for donation. And in order to assure the safety of the plasma, some countries rejected the parous females for the risk due to the risks of TRALI, while some others, for instance, tested for anti-HLA and HNA or even HPA antibodies. And in some countries, in some places, Also, a question to define was the donor compensation, because in some countries there were a truly competition with paid plasma collection sites that were also looking for a plasma from convalescent patients to support hyperimmune globulin preparation. And just as an example of those challenges that we all faced in the early days of the pandemic, let me share with you our experience for screening donors for plasma donation in our hospital in Barcelona. For instance, we managed to identify 104 volunteers with past history of COVID-19 infection. And in the selection process, we found that 23% of them didn't have peripheral veins suitable for a donation by apheresis. So we have to discard 23% of them. Also, we deferred about 7% of them due to other reasons. So in the end, we had 70% of the initial people that were finally tested for the donation process. And we found that in spite of having a history of COVID-19 compatible symptoms or disease, actually we found, almost 20% of them that didn't have antibodies. And also because at that time we were testing parous females for HLA antibodies, we found 5% of them with positive antibodies that had to be deferred. In the end, so we had 47 donors that were scheduled for plasmapheresis donation. However, 10% of them had problems during the collection or didn't show up. So in total, we have 42% of donation of more than of the 100 volunteers that we screened. So imagine that the challenge that represents to collect enough plasma to meet the demand that we had at that time. We have analyzed the challenges that were common for high, low, and middle income countries. Now we can focus in just challenges that were particularly found in low and middle income countries. Well, in those countries, you know that unfortunately there is a chronic transfusion deficit. The stress, the additional stress to the system that represent the collection of plasma was really a real challenge. Also, the problems that those countries have with donor recruitment policies, that actually it varies, those policies vary widely, but unfortunately many countries still the donor recruitment policy is based on replacement and paid donors. Also, in those countries, the general organization is a decentralized transfusion services. So with different places collecting that blood donation and plasma donation, that represent additional difficulty in order to
Please check your audio. I'm sorry, but the jack maybe... No, we don't hear you. No. No way.
Do we lose the connection?
No. Please try to move like before. It works.
Pardon?
Yeah.
Can you hear me? Can you hear me now?
Not very well. Just a bit.
Just.
No, I'm sorry. I'm sure that it is the jack inside the PC that it is not so good. So try to move
We cannot
again,
hear
or
Mikel.
we go forward.
And I try to. Can you hear me? Hello?
Yeah.
Hello?
Okay.
Mikel?
Yes?
Now we can hear you.
Okay. Let me try again to share.
Just the last slides.
Yeah,
Yeah, from there. Yeah.
from there. Okay?
Now we can hear you in Rome at least.
Okay, thank you. Well, I was mentioning the problems that those low and middle income countries have with the specific for those countries. I was saying that donor identification is also a challenge because the reduced number of registries and databases, also the restricted access to existing database, and also the limited internet access that in some countries occurs. Also, I have already mentioned the difficulties that in donor recruitment due to the model of the model of voluntary non-remunerated donors is poorly implemented in those countries. Also, donor eligibility can be an issue due to the limited access to laboratory screening tests. And also that makes, for instance, that very often parous women are deferred from donation, limiting the donor pool. And also, many countries, unfortunately, they have restricted access to apheresis technology for donation. And obviously, also the access to pathogen inactivation technologies for plasma is not in use in many low and middle income countries. So we could conclude that convalescent plasma was one of the first therapies used for treating COVID-19, that the recruitment and qualification of sufficient donors to meet the growing demand of those days was a huge challenge in the blood centers around the world, and that those challenges in general were even higher, unfortunately, for low and middle income countries. And that's all. Thank you very much for your attention.
So thank you very much, Dr. Lozano, for your interesting and important data that you could share with the public. So just to remind the participants that there's a question and answer box that you could fill in, and at the end of the session, we'll answer as many questions as we can. So the next speaker will be Richard Ford from the Council of Europe. So please take the floor then.
Hello, can you hear me okay and can you see me okay?
Yep, both.
Excellent. So thank you very much. Just firstly, I'd like to thank Vincenzo and all at the Italian National Blood Centre for the invitation to speak here today at the World Blood Donor Day Scientific Symposium, and just to take this opportunity to extend a large thank you on behalf of the EDQM to all blood donors who, despite challenging circumstances over the last year, have continued to give generously to ensure the availability of life-saving treatments for the patients who need them. So I'm going to attempt to share my screen, so please tell me if there's any issues. Hopefully you'll be able to see my screen now.
Yep, perfectly.
Excellent. Okay. Okay. So my name is Richard Ford. I'm a scientific program manager for the EDQM of the Council of Europe in blood transfusion. And I'll just give a very brief presentation on some of the activities performed by the EDQM in blood transfusion, and touch on some of the main topics to be discussed during the course of this symposium. So obviously the convalescent plasma as is the topic of this session, and importantly on donor protection and horizon scanning, and looking forward to emergency preparedness and contingency planning to ensure the continuity of blood supply. So I'll start by firstly giving an important place to start is what exactly is the Council of Europe and the EDQM. So the Council of Europe is the EDQM's parent organization. It's founded in 1949 and its headquarters are in Strasbourg in France. And the Council of Europe is made up of 47 member states, which covers over 820 million citizens. It's the oldest pan-European organization dedicated to fostering cooperation in Europe, and it promotes democracy, protects the rule of law and human rights, a lot of which applies to the blood transfusion setting. The EDQM itself is the European Directorate for the Quality of Medicines and Healthcare. It's a Council of Europe Directorate, which was established based on the Convention of the Elaboration of a European Pharmacopoeia through a partial agreement in 1964. And the mission of the EDQM is to contribute to a basic human right, access to good quality medicines and healthcare. So to give an overview of the Council of Europe's blood transfusion activities, the activities themselves are led through the European Committee of Blood Transfusion, the CDPTS, for which the EDQM acts in the capacity of technical secretariat and has done so since 2007. And the work of the CDPTS is based on three main pillars. So voluntary, non-remunerated blood donation, mutual assistance, and the protection of donors and recipients. And there are a number of activities which are coordinated by the EDQM through the CDPTS. And I suppose the primary activity is developing legal instruments, technical standards and policies, and the guide to the quality and safety of blood and blood components, the blood guide, which is developed through a working group of the CDPTS, the GTS, is now in its 20th edition, and also contains good practice guidelines for quality management systems as well for blood establishments. A second activity is through monitoring data and best practices through annual surveys of activity data. And also there's operational activities supporting blood establishments in implementing technical standards and European legislation. So audit programs, external quality assessment schemes, among other conferences and training programs as well. So as well as those activities, there are a number of working groups to which the CDPTS nominate. And I just wanted to highlight some of these working groups today, including the TS120 on convalescent plasma, the TS93 on plasma supply management, and the BSEP working group, so the blood supply contingency and emergency plan. So two CDPTS nominated working groups, which I suppose are of particular relevance for today. The first one being the TS120 working group, which was nominated through the CDPTS towards the end of last year, and will commence its activities this year to investigate the various issues related to convalescent plasma donors. So building on the experience of the COVID pandemic and the COVID convalescent plasma and the ongoing work that's performed in COVID convalescent plasma, will look to investigate those issues, but also, where possible, develop generic guidelines for donor eligibility criteria for convalescent plasma. Another CDPTS nominated working group is TS93, which is a longer established working group created in 2013, which looked at plasma supply management with the main objectives to develop tools for assessing self-sufficiency in member states, to develop tools or give advice to achieve a higher degree of self-sufficiency, to use plasma of the required quality to its full potential, to achieve sustainable and safe plasmapheresis, with a particular focus on donor protection, and to provide evidence-based data to support revisions of the common technical specifications in the blood guide. So TS93 as a core group works with or has worked with key stakeholders involved in the field of blood transfusion and in PDMPs or in plasma-derived medicinal products. So the EBA, the EPA, IPFA, PPTA and PLUS. And just something to highlight to you, which I thought was important for today, is the Plasma Supply Management Symposium, which was held jointly organised by the European Commission and the EDQM in January 2019, again with a focus to discuss the obstacles on creating a strategic independence of plasma for fractionation in Europe. And again, one of the most important things to discuss donor protection, safety, selection and management. So I just wanted to highlight this. The proceedings from this symposium are on the website, along with recommendations to key stakeholders as well. Finally, I just wanted to highlight another project which is ongoing at the moment at the EDQM, is the Blood Supply Contingency and Emergency Plan, the BSEP project. And the overall objective of the project, I suppose, no more relevant than ever, given the year to 18 months that we've just had, is contributing to strengthening national and European level plans to ensure continuity of blood supply in emergency situations or responding to disruptions. It's ongoing at this moment in time, and from this group we look to develop recommendations for stakeholders and develop model preparedness plans where we can as well. I think one thing that has always been evident in emergency situations or where faced with challenging situations is, particularly as we've seen with the COVID-19 pandemic over the last number of months, is that blood donors have always continued to give generously and to support the blood services in emergency situations. And I think that's just a point that I'd like to finish on. So that's my last slide. I'd like to thank you for listening, and I'll hand back over to the moderators. Thank you.
Okay, well, thank you very much, Dr. Ford, for your presentation. So we will like to continue with a presentation from the European Commission. Stefan van der Spiegel, the floor is yours.
Thank you, Cynthia, and thank you, Nicola. And also I would like to start at this occasion first also from the European Commission to give a warm thanks to all the donors that are supporting our initiatives in our sector and make the use of transfusion available. Also, this is a message that my commissioner at political level has strongly made in those last days. So in the coming 10 minutes, I would like to take you through what we have been preparing and the activities that the Commission has been playing in the field of convalescent plasma. Can you see my slides now, Cynthia?
Yes, please go
Okay,
ahead.
I'll put them full screen. Okay, good. So maybe start first with a situation of the EU, and I think as in other parts of the world, we were strongly hit by the pandemic in a couple of waves, as from spring 2020. That also meant that rapidly we had a group of recovered patients that could be were available to provide antibody-rich plasma. The services, the blood service across the European Union actually started quite quickly with collecting plasma from donors and using it in patients, both in clinical trials and outside trials, because it was known quite early on that it's a safe therapy, and there was, of course, no alternative therapy available. So for us at EU level and as the European Commission, we saw here the need and the potential to make sure that the data were gathered well from collection and use. Yes.
Excuse me. Can you close the gallery on Zoom because it's overlaid on your slides? Yeah, that one.
Is this better?
Yeah. Okay. Move it up.
Yeah, it's
okay,
in the
perfect.
corner
Thank
now.
you. You can.
Welcome.
Thank you.
Okay. So for us, I think our aim always has been as European Union, as Commission, to make sure that the data are gathered well and that we take the best learnings out of this real life experiment. And maybe also just to show on the right hand, the flow we're talking about is the flow number 1, where the plasma is collected for transfusions for in different settings. But we also have in Europe a couple of pharmaceutical companies who also are manufacturing the derivatives out of the plasma, and so there was also collection for those for that pathway. So as the European Commission, we basically have developed 4 supporting actions in general. First one is to set some guidelines on how convalescent plasma can be best collected and used. Second one is to support the research in the field with dedicated research funding that we have available. So overall, there was more than 7 million euros of research granted into the field of plasma, both convalescent plasma for transfusion and hyperimmune plasma derivatives. Then we also, and I think that was our main initiatives that I also want to say most on, we have been developing a dedicated database on that to collect the data on donation and on outcome. And the last part is that we've also been funding the blood services across the EU to increase their collection capacity. I won't come back to that further later on because I think it's a bit less relevant in this global setting, but there has been quite some initiatives ongoing to make sure that the blood services across the different EU countries had the possibility to increase their plasmapheresis collection capacity so that they would have quicker and more convalescent plasma available. This is an investment at EU level that we are also considering useful in a broader political context, in the broader context where we have, as EU, not and coordination. It's not the European Commission itself that does these initiatives. The guideline we developed in close collaboration with the blood authorities and with the European Centre for Disease Control. The database we developed in close collaboration with the European Blood Alliance that we will hear later, and with colleagues in DG Digit, which is more on initiatives. The funding came a lot from our research envelopes, so Horizon 2020 it's called, and the funding for increasing the capacities were coming mainly for a dedicated envelope for emergency support. So first, a word on the guidance. So this is guidance that we developed, as said, in very close collaboration with the European Centre for Disease Control, but also with the blood services and with the national blood authorities. The aim of this guidance was to make sure that, as we know that as blood service we're immediately starting to collect convalescent plasma, that is done in a good way, and where possible also in a harmonized way that would allow for common collection of data, both on the collection, the donation side, and on the utilization side, so for future learning. So the guide has also been updated regularly to follow the science. I think this currently this is the 4th edition on our site. So it includes a state of play of what we know, very much as Cynthia summarized at the beginning. Then it also includes specific elements on how to look at donor eligibility, post-infection, post-vaccination, how to look at collection, how to do the testing for the SARS-CoV-2 antibodies, and also there's a part on the distribution and use with a focus on early application and on the use of high titers. And there's also in there, and that's the link to the next part, there's in there also some guidance on how to collect data, both from on the donation side and from the utilization side. So data that allows afterwards to have a common view on the effect of the therapy. And that brings me to our database that we developed in very close collaboration with the European. They're set up in a couple of levels. There was a registration level that we also worked together with EBA on. Then there's a donation module to bring all the donation data together. Then there was a treatment module. So all the data were brought together in an anonymized way. And then there's also some public dashboards available on it, but so the data, and I will show some of it, can be publicly partly consulted, but the real analysis is with the European Blood Alliance. I'll come back to that also later. So this has been developing process. It started already in April last year, but there are more and more modules were added and more tools are available. So at the moment, we have around more than 130,000 donations registered in there. We have of them around 28,000 were qualified for transfusion according to the local protocols. We have overall 113 blood establishments from 25 countries that have registered, and that includes EU, but also UK and Switzerland. But the upload of information was so far only done by 55 establishments from 16 countries. And we have, and that's a much lower number, we have information on around fully uploaded around 750 COVID patients from 8 countries. Only there's clearly a discrepancy with the number of donations, so we are expecting a take up by the blood services in the coming weeks and months to increase that number and to increase the learnings. So a couple of just to show you a couple of the dashboards that you can also find online. So this is one on the donation data, and you can in there go into the different countries and look into the data. You can also make basic analysis like the gender distribution, where you see there's more female donors, the body mass index, the donor age, the day since the symptoms have stopped and up to the donation. So that's a part of the database that can be also publicly consulted on. And then we also have dashboards on the patient side, but as mentioned, much less patients. Also, one of the learnings, I think, is that there's an extra step, a gap to get the data really back out of the clinic. This is no longer just the blood services that need to get the data. They need them also to get back from the clinic. But it gives you already some views on treatment settings in a clinical protocol, a clinical trial, or monitored use. Also the the use of the different diagnostics for the disease. So there's a couple of elements again that can be browsed through those dashboards. Then, as I said, the real analytical work on the data is actually undertaken through a research-funded project that's called Support E, and that is led by the European Blood Alliance together with 11 national or local blood services. On the left hand, you see the different work packages they work on. One is clearly analyzing the database, but they also will work on producing data sets to add to the database. They will also work on the potency assessment. I don't think I have to repeat this. Cynthia already presented as well, I think, where we are with CCP, what we know and what we would like to know more. For us, then maybe to end, what are the messages we got out of this experience is A, that fast action is possible, but we really need good collaboration. And I think we have that in Europe between professionals, authorities, the Commission and the European Centre of Disease Control, who I think throughout this whole exercise was really a very respected partner and provided a lot of inputs. I think it's also important at this moment to thank the role, the European Blood Alliance and their central role in coordinating the professionals and the blood service. I think that was essential to make this move forward. So as said, to understand and trust the medical professionals was key, and I think EBA played a central role in that. We also, for ourselves, learned better out of this that there are quite some possibilities we have in the European institutions to support this kind of initiatives, both on the research side, on the digital side, and in case of need for emergencies. And obviously, it's also clear that digitalization is driving healthcare. So in that context, this whole experience on the convalescent plasma database was really a valuable experience that for us, as you might know, we really want to take into account and want to take those learnings with us as we are at the moment revising our legislation in the European Union on blood and blood components. So I think several of the learnings out of this exercise will be very useful for that. So I think that was in short what I wanted to present to Cynthia and Nicola. Thanks for your time.
Thank you very much. I think it's now my turn to introduce, if I got it right now, my former colleague Yu-Yun, Marie-Yun Thi, from WHO, Geneva headquarters. Let me repeat again, she's team lead of the blood and other products of human origin at WHO, Essential Medicines Department, and she will give us her speech on WHO recommendation on collection and use of COVID-19 convalescent plasma. Please, Yu-Yun, you have the floor.
Thank you, Dr. Magrini, for the introduction. So once again, I would like to thank the Italian National Blood Center for giving a chance to WHO to share our interim guidance, consists of recommendation on collection and use of COVID-19 convalescent plasma. So I will share my screen now. Sorry. Yeah, I will start. So in this presentation, I will briefly explain reasons of using of convalescent plasma for treatment of infection, as well as collection and also use of convalescent plasma for COVID-19 treatment. So as we all know that convalescent plasma is plasma collected from someone recovered from infection through whole blood or plasma apheresis collection. And convalescent plasma contains neutralizing antibody, so make it different from vaccine. The plasma from COVID-19 recovered patient has antiviral effect because it has specific antibody, or we call it neutralizing antibody to SARS-CoV-2 antigen. The convalescent plasma has also immunomodulator effect, which involve the complement cascade, cytokine regulation, and also other factors. And other immune pathways, such as antibody-dependent cellular cytotoxicity, complement activation, or phagocytosis, are putative mechanisms through which convalescent plasma might exert its therapeutic effect in patients with COVID-19. And to develop the interim guidance on use of convalescent plasma, WHO cited some studies which is available during the time when we developed this recommendation. So we developed this recommendation in October last year, and the recommendation was published in February 2021. So in this slide, I would like to describe some study that we cited. So one study is from the Cochrane database of systemic reviews in October version. So it mentioned that the definitive evidence for safety and efficacy of COVID-19 convalescent plasma currently is lacking. And another study from Joyner et al. Said that some control studies have failed to demonstrate a clinical benefit. Another study, we quote that among patients with severe or life-threatening COVID-19, convalescent plasma did not result in statistically significant improvement. And reports from randomized controlled trials and several uncontrolled case series of use of COVID-19 convalescent plasma have suggested favorable patient outcomes. And another one is a meta-analysis of multiple clinical studies and retrospective analysis of a large database of protocol-driven but uncontrolled experimental use of COVID-19 convalescent plasma in the United States also demonstrated favorable outcomes. Various studies, including one involving treatment of 20,000 patients with COVID-19 convalescent plasma, have reported low rates of serious adverse reactions that were similar in type and frequency to those seen with non-immune plasma infusions. And there was also no evidence of antibody-dependent enhancement, a well-recognized effect in other viral illness. And positive historical experience with use of convalescent plasma to treat SARS and pandemic influenza further supports the plausibility of a clinical benefit in COVID-19. So based on these studies, WHO developed the recommendation on use of CCP for COVID-19 treatment through the interim guidance, which is issued in 17 February 2021. So this interim guidance actually is the third updated interim guidance in related to COVID-19 pandemic. So WHO recommendation on use of the CCP, the first one, WHO recognizes CCP as an experimental therapy for evaluation in clinical studies or as a starting material for the manufacture of experimental hyperimmunoglobulins. WHO recommends the use of CCP in randomized controlled trials. And in setting where patient randomization is invisible, structured observational studies similar to active arms of an RCT can be considered. And where clinical studies are not possible, patient outcomes should be documented and blood samples from donors and recipients archived for future characterization. This slide describes the criteria for acceptance of donor of CCP. I will not repeat this one because I think Dr. Lozano has mentioned the criteria of donors for the CCP donation. And in this slide, I also would like to mention that plasma apheresis is the recommended mechanism to obtain CCP, and that neutralizing antibody titer measurement in the unit of CCP is really important to be done. And when giving CCP to the patient, there is a need to report the patient characteristics such as sex, age, and comorbidities, timing of therapy in relation to disease onset, administration of therapy, clinical and laboratory indicators of disease severity, adverse reaction, and time to hospital discharge. And furthermore, the interim guidance also advised to retain CCP samples when neutralizing antibody titer measurement is not feasible for future characterization, and later stratify outcomes from the treatment by the dose, including volume and titer of the CCP unit administered to determine the effect of titer CCP on the clinical outcomes. In this slide, WHO would like to advise that the use of reagents that have been calibrated against the first WHO international standard for anti-SARS-CoV-2 immunoglobulin. And this is to determine the binding and antibody activity of SARS-CoV-2 body. So by using this kind of reagent, we will be able to express the assay result in international unit and also could be able to reduce the inter-assay variation. And I think this is just a short presentation. And to close my presentation, I would like to say that historically, convalescent plasma may be effective for treatment of virus infections early in the course of the disease and in certain amounts of antibody level. And it seems that only CCP with high titer of neutralizing antibody is benefit for treatment, and definitive evidence for safety and efficacy of CCP currently is lacking. Therefore, further clinical studies is needed. So until this time, WHO recommends the use of safe CCP under the clinical studies. So thank you for your attention. Back to the moderator.
So thank you very much, Yuyun, for your interesting and very valuable presentation. And I still think that collaboration is really vital to come to these conclusions. So the last speaker will be Pierre Thibaudier from the European Blood Alliance. So Pierre, the floor is yours.
Thank you very much, Cynthia. Hello to all. Good morning, good afternoon, good evening. I'm very happy to be with you today, and I would like to thank Vincenzo and colleagues from the Italian National Blood Center for giving me the opportunity to present today. And I will be now sharing my screen. Do you see it right there or not?
Yes. Not now.
There we go.
First, yep.
Yeah, okay.
Perfect.
Okay.
Perfect now.
Very good. Okay, so, and if I switch this, is it still okay now?
Nope.
No.
No, it's not. Okay. I'm getting it back the other way around. Better like that?
Yes.
Okay.
Okay, well, thank you very much. So, I'll be presenting today the European Blood Alliance and Support E perspective regarding COVID-19 convalescent plasma. I would like to start by briefly presenting the European Blood Alliance. I would like to thank Stefan for his very kind words with regard to the European Blood Alliance. It's an association of nonprofit blood establishments across Europe, 26 members, 3 observers, and all members of EBA throughout Europe share very important core values: patient care, donor safety, voluntary non-remunerated donation, openness through information, and sharing and excellence through evidence. As we move through the COVID-19 crisis, and early on, EBA was very active, and in particular, sharing information and best practices with its members, with other associations, and in close collaboration with the European authorities, DG Santé, ECDC, and others. Also supporting EBA members in the effort to ensure continuous and timely provision of safe and high quality blood components. Also supporting our members to protect blood donors and protect the staff. And this is today's topic, support and promote the evaluation of convalescent plasma as a mean to treat COVID-19. So a lot, and as highlighted earlier, blood establishments among EBA members embarked into the collection and provision and issuing of convalescent plasma. And the European Blood Alliance was very much active in two domains, one very well covered by Stefan, so I'll go quickly through this, which is the contributing to the setup and run of the EU COVID-19 convalescent plasma database, which is this open access database collecting donation as well as treatment information with the aim to consolidate the EU evidence on the safety and effectiveness of convalescent plasma. Also, several members of the EBA, with EBA as a coordinator, put together and proposed a project to support high-quality evaluation of COVID-19 convalescent plasma throughout Europe. This was funded by the European Union, the Horizon 2020 program, and this program has started, and I will be now describing several aspects of this project, which aims to assess, to support, and to come up with recommendations as to the use of convalescent plasma in the current health crisis, but also, importantly, in subsequent crises involving novel pathogens. This project puts together, as Stefan showed, a large number of blood establishments with corresponding clinical teams throughout Europe. You have the list here. And we also have a participation of a larger number of blood establishments throughout Europe, which will be contributing to the recommendations. As these European funded projects, it's divided in work packages, which I will now be detailing with you. A first work package was there to basically set the scene, assess what's going on in Europe with regard to convalescent plasma, to conduct, to make evaluations of what's going on with the clinical programs and defining best practices. And doing so, produced preliminary clinical trial guidelines and assessment criteria. Also, regard to monitored access, which is compassionate use, but in a monitored way, and we really would like to favor this throughout Europe, assess upcoming and ongoing clinical trials, and also make a pre-selection of clinical trials and monitored access studies to be further supported. The second work package is there to support, and through financial support in particular, high-quality clinical evaluation and producing data sets for the inclusion in the EU database. Here, there is a lot of activity surrounding promoting clinical trial and monitored access data uploading to the EU CCP database, as well as moving forward into clinical evaluation, into the support which can be brought in that regard. And basically, today's priorities, as viewed by Support E and by others as well, and as highlighted earlier on, is that we believe we need to intervene very early in vulnerable patients. There needs to be a specific focus on patients unable to mount an endogenous immune response to SARS-CoV-2. And just to show some data here, and I showed earlier, is by and large, when one looks at hospitalized patients, but that is not the very early intervention, overall, and you can see this in this forest plot here, there has been no clear evidence of efficacy of convalescent plasma in that specific setting. However, and in line with what's been seen with monoclonal antibodies and also in other infectious diseases such as Argentinian hemorrhagic fever, very early interventions, that is in the days right after symptom initiation, there might be here a place for convalescent plasma, as highlighted in this trial by our Argentinian colleagues who showed in this study by Lipster et al. That this very early intervention in vulnerable patients, being elderly patients mainly, convalescent plasma could basically halve the risk of worsening towards a form of disease which would require hospitalization. Importantly, it was seen in this trial that there was a dose effect, and that high titer plasma was important. And in that case, there was a 73% reduction versus only a 31.4% reduction in worsening of the disease with lower titer convalescent plasma. A more recent trial, which is not in this forest plot, which is a German study called CAPSEED, was recently made available and also made, I think, an important point with regard to the dose and to the fact that we need to go to high titer plasma. And showed here that when one looked in this randomized study in patients who had received a very high titer plasma, there was a significant improvement, statistically significant, both for clinical improvement, discharge from hospital, as well as overall survival. So this issue of high antibody titer plasma, I think, is indeed very important. And we can see this, and it has been shown that possibly vaccinated convalescent donors could be the ones we should use, because as shown here in this publication, the vaccinated convalescent donors, convalescent and then vaccinated, have not only very high titer of antibodies, but also have what we call cross-variant neutralizing antibodies against variants of interest. And also, and showed earlier on by Cynthia, there has been a lot of interesting data suggesting that in immunosuppressed patients unable to mount an immune response, and for which there is no access to vaccination, this should be importantly looked at. So all this is bringing us to suggest with Support E that we should be into very early outpatient intervention in vulnerable patients, and in particular with a trial called COVID-19 and in preparation, which will be assessing this approach both in elderly and less elderly patients with comorbidities, but also, and importantly, in a distinct immunosuppressed patients cohort with several countries, which you see here throughout Europe. Secondly, when you think about the specific focus on patients unable to mount an endogenous immune response, well, the COVID-19 trial will be assessing these patients early on, that is, in high-risk patients before they require hospitalization. But also, we are currently thinking about moving into assessing again in a randomized fashion, this time in hospitalized patients, the interest of CCP. A subsequent work package is collecting and monitoring and analyzing the data into the database. So this was very much shown by Stefan, so I'll just go quickly through it just to say that we are generating a lot of data, both with regard to donations. Just an example here, the dates of donations throughout the last year, the year at the donor age at donations, neutralizing titer, which I think is an important issue and for which I'll be showing some data subsequently. From the recipient side, so we have less data currently, working very hard on this issue. Some issues with GDPR, which is slowing things a bit, but we'll be moving, I'm sure, forward, I hope, very quickly. With here also data, just as an example, the age range of recipients or the number of days between the onset of symptoms and the first transfusion episode. So it's just to show that we are generating a lot of data, which will be very interesting now to start analyzing. The next work package is looking into improving plasma potency assessment. This is very important. We've sent out a questionnaire to a large number of countries in Europe, and we have, as expected, a high diversity of assays. We are embarked into a calibration of these testing, and we've been able, notably with regard to measuring SARS-CoV-2 neutralizing antibody titers. So this is a functional assay, and this was done in 11 countries, 16 laboratories. And this, by exchanging samples, we were able to calibrate titers with international units, as described earlier, and resulted in a 50-fold reduction in interlaboratory variability. And this will allow for direct comparison within trial results. And we're doing the same thing with regard to measuring directly antibodies and ELISA assays, which is currently ongoing. The last work packages are to develop recommendations and to prepare for the future. So this is more in front of us, but just to say that the systematic living review, which was highlighted earlier, is done with the support of Support E and is providing highly valuable information as detailed earlier. One issue which will be important is to position convalescent plasma with regard to other approaches of passive immunotherapy, be it monoclonal antibodies or hyper-IG. And so it's issues of comparing efficacy, risk for immunoselection of variants, administration modalities, availability and cost are going to be important to consider. And just as a start, to highlight that convalescent plasma is the only passive immunotherapy approach readily available early in a crisis. It's basically immediately available, which is obviously different for the other approaches. It allows for a fast turnaround in response to the emergence of variants of concern, and that's important to consider as well. And also it contains IgG, IgA, and IgM isotypes, and could be important when comparison to hyper-IgE, which contains only IgG, so issues for the future. All this needs to be disseminated, to be exploited, and to be communicated. And this is an important work package. And I would like to take here the opportunity to thank Livia and Vincenzo, both for the strong participation to support it, as well as obviously today's event and giving me the opportunity to present. And lastly, but importantly, a project manager by the European Blood Alliance, and notably Catherine Hartmann, Executive Director, and Gaia Mori, a project coordinator. So I'll stop here. And so just to say that as we see, once again, we see that blood donors are on the front line. And so together, we thank all blood donors worldwide for their most precious and never-ending commitment to patient care. Indeed, they're keeping the world beating. Thanks a lot. And I'll finish there.
Well, thank you so much, Pierre, for this very interesting, impressive talk. Also, evidence on collaboration within the European Union. So I think it's still very important to have this kind of collaboration. Also, funding is also very essential, of course. So we're now, I would like to thank all the speakers again, and we now go to the question and answers part. I don't know if there are any questions, but please put it in the question and answer box. Just first question from me to the speakers, that I have a great wish to have like a worldwide collaboration, like a kind of platform on sharing information, sharing data, etc. Would it be possible to start such a collaboration, you think, including all those important societies and organizations, but also outside EU, WHO, of course, ABB, ISPT, but also societies from Africa and also Asian societies? What do you think? Is that doable, possible? Would like to hear your thoughts.
Well, I'll be maybe happy to start. I would say that that's absolutely essential. It's something we should be working on. We obviously need to be organized at different levels, and what I showed at this moment was what we're doing currently at the European level. But obviously there are interactions ongoing, and just to mention the calibration efforts, which is ongoing, is clearly to be done in close collaboration with what's going on at the WHO and presented earlier on by Jeroen. I think these are essential aspects and need to be invested in strongly.
Yeah, I would like to support what Pierre said, because as you may still remember my first presentation, that the strategic objective number 6 of the action framework is actually building the collaboration with other partners. Thank you, Cynthia.
Maybe I can add from the European Commission side that that's definitely the overall spirit in which we like to work. I think also if I presented on the guidelines for our EU blood attachment, that those are also well aligned with the WHO recommendations. The database presented is also is open for blood service in non-EU countries. Overall, I think whatever we do and make available from EU side and sources is usable and available to be used broader also.
And just to support that, I completely support everything that's been said by the other speakers as well. I think that there's definitely a need for collaboration and in some way to look at how we can support the standardization of the data that's collected as well. I think it's very important that we're all kind of operating on a kind of a standardized way going forward as well. So 100% support the other speakers as well.
Well, thank you. There is a question from Vincenzo. Maybe we
yeah, I was just I was just know if someone want to comment about the fact that if there is any room for the use of immune globulins, of purified immune globulins from plasma. And as Pierre said, of course, it seems that the nicest source for this is plasma from convalescent and vaccinated donors. So my question is, would this plasma become from the blood establishment, and what will be the role of blood establishment in that? Because it means appropriate selecting donors for collecting plasma and producing immune globulins. This is something relevant for the job of the blood establishment. And I don't know if someone wants to comment on that, because...
So I think I'd like to respond to your question. So from the WHO point of view, I was thinking that this kind of mechanism need, again, the education to the blood donors, and also maybe finally we also develop the informed consent from this kind of donor. The plasma for, for the use to prepare the hyperimmune globulin product. Thank you.
Maybe I can perhaps add to that. Yes, indeed. I mean, as we're thinking now about moving towards convalescent vaccinated donors, the fact that these same donors could give plasma for direct transfusion use as well as for hyper IgG, I think is obviously, I would say, is the way to go, and would be an important, probably addition to the potency and cross-reactivity of the antibodies contained into this hyper IgG. Lastly, I would say that regarding hyper IgG, and I think it's currently missing, we do need, and I understand it's ongoing, and I think and particularly in the Netherlands, that we should be investigating early administration of these hyper IgG, just as we're doing currently with convalescent plasma and has been done with monoclonal antibodies.
Okay, well, thank you all, because we're running a bit late. Well, I would like to thank all the speakers. I would like to thank all the participants, and I would like to thank my co-chair, and then end the session and give the floor back to Vincenzo. Thank you all.
Yeah, thank you very much.
Thank you.
It has been really a very interesting session, nice presentations, and a lot of things that we can bring home as messages. And so I would end this first session, and we have now time for a break for a lunch, and we come back at 10 minutes past 1 p.m. for the second session, which is devoted to country perspective and initiative on contingency planning to ensure adequate blood and blood component supply in different situations. So thank you again to everybody attending the session, and see you later.
Thank you. Thank you to the moderators. Bye.
Hi, thank you all. Thank you, CNS, for bringing us together.
So, bye.
Thank you. Bye.
Thanks as well. Thank you.
Well, good afternoon to everybody. And we are at the beginning of this second session. Very interesting title is Country Perspectives and Initiatives on Contingency Planning to Ensure Adequate Blood and Blood Component Supply. I will be co-moderating this afternoon session together with Dr. Yuyun Mariuningsi. And I would like just to have one slide, just to introduce the topic and to demonstrate how, in my opinion, it is so important to have a country perspective and a region perspective and a wide perspective on initiative in contingency planning. And the slide demonstrates something very clear. It is the comparison between the red blood cell produced and transfused in 2019 and 2020, and you can, in Italy, and you can understand the difference is just what happened due to the pandemic. And you see from this simple slide that in some instances, and pandemic was one, or you don't have the blood when you need, or you don't have the need when you have the blood. And so how to ensure a constant, regular, adequate supply of blood and blood components in timely available way, it is one of the utmost important things that we have to guarantee to our system. And having introduced in this way the session, I would like to give the floor to my co-moderator, Dr. Yuyun Mariuningsi. So please take the floor.
Thank you, Dr. De Angelis, for introducing the topic of this session. So good morning, good afternoon, good evening to all the panelists and also to all the attendees. So we are happy to have 4 speakers for this session. So I would like to introduce the first speaker. So the first speaker is Dr. Aparna Shah. So Dr. Shah is our Regional Advisor for Blood and Transfusion from WHO Southeast Asia Regional Office. So Dr. Shah will share with us the regional experience on contingency plan on blood supply system. So Dr. Shah, please, floor is yours.
Thank you, Yuyun. Thank you, Chair and Co-Chair. I can start sharing my slides now, can I? Can you see my slides?
Yes.
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 Can you put it in the slide, Dr. Shah?
Yes, it is in slide mode. So
yeah, we can see it, yes.
Yeah. Good morning, good afternoon, and good evening. Namaste and Assalamu alaikum. At the outset, on the behalf of WHO Southeast Asia Regional Office, I would like to thank organizers for inviting us. In my today's presentation, I'm going to cover blood transfusion services in Southeast Asia region, impact of COVID, success stories, and role of WHO in supporting blood safety initiative. So here you can see our 11 member states. We occupy 6% of land area, 26% of total world population. So as all of you are aware, timely affordable access to safe blood is essential to ensure UHC. And UHC is our regional director's flagship program. Availability and accessibility to safe blood varies in countries and within the country. In our region, blood shortages are more pronounced in rural and hard to reach areas, and outbreaks poses additional challenges on blood safety. So here in this slide, you can see blood is a vital healthcare resource. In developed country, where we use blood mostly for surgery, massive trauma, hematological malignancies. In our part of the world, we use it mostly for pregnancy-related complications, severe childhood anemia, trauma, and now, of course, in many countries for surgeries and malignancies. Blood can save lives, but at the same time, it could be vector for harmful infectious diseases. And now, new pathogen may pose additional threats to safety and availability of blood supply. So WHO has been always at the forefront of movement to improve global blood safety. 1998, we established GDBS because we wanted to know what member states are doing related to their blood safety initiatives. We started collecting data. They are still submitting us annual data to GDBS. Then in year 2000, two major events happened. One was a not regional, global blood safety strategy. And also we celebrated World Health Day and our theme was Blood Saves Life. In 2005, World Blood Donor Day, we started celebrating as public health campaign. So why I'm saying all of this? Because whether we talk about a global strategy for safe blood, which came in 2000, or the new form of the strategy, action framework, which Yuyun presented in morning, action framework to advance universal access to safe blood. In brief, it's about nationally coordinated blood transfusion services, VNRDs from low-risk populations, testing for all blood in a quality assured way, reduction of unnecessary transfusion, rational use of blood, component separation to make best use of this vital product, and quality encompassing every aspect. So GDBS I've already talked about. Annually, member states are submitting data to us. So this presentation is based on the data submitted by the member states. Now coming to the region. So most of the member states, they have unit in MOH with responsibility for governing all activities related to provision and transfusion of blood and blood products. National blood policy, which is very, very important document. I'm happy to say all 11 member states have a blood policy document. Most of the member states have specific legislation or other legal instruments covering safety and quality of blood and blood products. International agencies, organizations, institutions, NGOs are providing vital support to blood services. The region has in recent years made steady and sustained progress. I'm very happy to say all 100% of donated blood is screened for TBIs. The proportion of blood donated by voluntary donors is more than 80%. I must say it's around 84%. Between 2013 and 2018, the region increased blood donation by 2.37 million units, third highest increase globally. Number of voluntary donations increased significantly. Most countries have national guidelines on appropriate use of blood. In this cartoon, you can see we are collecting 16 million units per year. The 11 countries in Southeast Asia region reported the collection of 13 to 15% of global blood donation. But in the first slide, I have said that these countries represent around 26% of global population. So we are not collecting enough. Several social myths continue to hamper not only voluntary donations, but also blood transfusion acceptability. So in this slide, you can see Maldives, Thailand, Sri Lanka, they are collecting much more than 1% of the total population, the blood donation. Indonesia is slightly above 1%. Bhutan is also slightly above 1%. India is reaching 1%. But we need other countries also to reach at least 1% of total population who should donate blood. Then regarding voluntary donations, overall it's 84%. However, Thailand, Sri Lanka, BPRK, they're all collected blood is voluntary donation. India is 85%, Indonesia is approaching 85%, Myanmar 88%, Nepal 87%. Around 47% of blood units are converted into blood components. Now, very relevant point, I said in the beginning that 100% of the blood is being tested for TTIs. However, quality assurance of testing is questionable at places. Supply of reagent and kits is erratic at places. So I would like to highlight 11 M's which are needed for a very, very efficient blood transfusion service. Man, human resource, machinery, good equipment, material, reagents, methodology, standard operative procedures, protocols, management, motivation, M&E, maximum containment, matrix, that is network, maintenance of expertise, that's trainings, retrainings, and above all, money. If you have all of this, BTS, we can assure good services. So I will not go through the whole slide, all the points. So in brief, I would say we have a good system, maybe at tertiary care level. In some countries, it's really good. Some countries, not so good. They are working in direction for making the services better. So linkages of BTS with health services is missing at places. I said all countries have national policies, but not properly implemented in certain countries. Legislation and regulations are not functional to control fragmented systems at places. So in fragmented system, controlling quality is very, very challenging because it fails to understand needs of practitioners and patients. Transparency in BTS and mandatory centralized reporting of the source, volume, and type of blood donated to blood banks is important to calculate the demand and ensure availability and accessibility to safe blood. Data management and information system we can improve. I would not say it is 100%. Strengthening BTS network, it is important to understand the demand and extent, as I said in my previous slides. Hard to reach areas, we need to have the storage centers, but some storage centers only exist in Maine. Quality control is a very, very important part. We are trying to support EQUAS with help of our collaborating center, NRL Australia. We are trying to support national EQUAS system, but we need to work more on that. Procurement of good quality equipment and reagent is not regular at places. So better timely clinical management can avoid need of blood transfusion. As I said in my previous slide, postpartum hemorrhages, childhood anemia, if we can control, if our health system is better, the need may go down. Hemovigilance is almost gradually starting. Long-term implications of blood transfusion are not known at every place. Now coming to impact of COVID-19, it's the same across the globe. So we are no exception. Transfusion services are facing several challenges during this pandemic. WHO developed one guidance document before COVID that was protecting the blood supply during infectious diseases outbreaks, and one after COVID pandemic, which we update time to time. You can see on your screen. So these documents were really very, very helpful to the member states in improving their blood supply. They adopted these documents as per their need. CRO member countries have adopted several measures to avoid blood shortages in line with WHO guidance document. So big emphasis was on public awareness campaign, providing passes and transportation facility to donors, appointments prior to donations to avoid shrouding, use of mobile vans to collect blood, changing the blood donation sites, use of social media, management of blood stock inventory to ensure no stockouts, development of deferral policies in line with WHO guidance document, and social distancing at the time of blood collection, use of masks, hand hygiene, and other IPC measures in line with WHO guidance document. So some success stories, you can see the flags. Bangladesh started social motivation campaign on social media, which received very good response. I will not read everything on the slide. Bhutan national guidance on maintaining a safe and adequate blood supply during COVID period was released. Armed forces supported by conducting blood donation drives. India, effective use of social media, again, provision of transportation, issuance of passes during lockdown, mobile vans for blood collection. Indonesia blood center started sending teams to home or neighborhood to recruit donors. Maldives blood services motivated people for blood donation through social media. Twitter campaign got big success. Myanmar, the blood center provided free transportation for donors in the same way like India. The student and police personnel donated blood and motivated people during crisis. Nepal, better coordination with Blood Donor Association, social media campaigns played a major role. Sri Lanka implementing an online registration system for blood donors. Then same way social media use, mass media campaign. Thailand, special blood donation buses and ad hoc blood donation venues. Timor-Leste, they collaborated with development partners, took technical assistance from WHO, National Red Cross established a blood donor collection center in their office. These are some pictures of best practices. So now the role of WHO, as I've already showed, in the beginning, my second slide, celebrating World Blood Donor Day, developing global strategy, revitalizing it, action framework. But after COVID, releasing the guidance document, then supporting on-site. It was initially on-site, now it is online assessments, and then initially it was online training, now on-site trainings, support to ECOWAS, NICOAS, guidance documents. WHO also helped member states in developing partnerships and collaborations. So this is our very rich website. You can visit, you can download any document you like to do. And on the screen, you can see a donor who's a doctor and a regular donor from Indonesia. With this, I will close. Thank you very much.
Thank you very much, Dr. Shah, for your presentation, which is indeed describe the situation of the blood services in the Southeast Asia region. So I think now we can move to another speaker. So I would like to invite Dr. Mauricio Beltran Duran. He is a regional advisor for blood and transfusion from WHO American Regional Office. Dr. Duran, please, time is yours.
Thank you. Good morning, good evening, good afternoon, everyone, for everyone. I share my presentation now. Can you see?
Yes.
My slide?
Yes, we can see.
Yes. I try in this slide to present the Organization of Blood Supplied in Latin American and Caribbean countries, PAHO region. The framework of the action for PAHO is given by the mandate of 41 member states and territories, and by the WHO framework about access, universal access of blood. We work through 4 strategies, such as integration of blood service in the national health system, self-service in blood with some percent, 100% voluntary, non-remunerated blood donor, and in quality management in blood service. And the last strategy is surveillance, hemovigilance, and monitor or evaluation. With these strategies, we to achieve, or this strategy to achieve universal access to blood and safe components in the region. Our region have a very important advance, very important excuse me. The region present very important results, excuse me, in testing and in donor selection. But the other side, we in our region present important gaps and challenges in the supply of safe blood, such as a low donation rate and voluntary donation with less than 50%, and the existence of small blood banks. We love efficient in collection and processing, and disconnect in many cases from the network of blood service is not connected with this small bank. For this region, PAHO support member states to improve access to blood by strengthening their national policies, plans, and analysis of costs of blood processing, and promote the centralization of processes to have a more efficient blood service networks, and that improved access to blood. In addition, we support the state members and territories in the activities of quality, safety, and use of blood to reduce the risk of adverse events or blood shortage according to the PAHO and WHO plans. There is a big difference between the availability of blood in low-income countries and in high-income countries in the region. As you can see in this slide, this gap generates blood shortage in normal times and can put the health system at risk in an emergency situation. Only 15 countries and territories have rate blood donation superior of 17 units per 1,000 inhabitants. Organized blood system allow maintaining of a safe and permanent blood supply. They also respond adequately in emergency situations because they facilitate coordination and communication between the actors of the blood system in the health system. PAHO gave some guidelines to prepare blood system for emergency situations. These are also applicable in pandemic to guarantee the safe supply of blood. These recommendations include coordinating mechanisms with the blood service network to identify banks with response capacity, with big capacity, to monitor inventories and blood needs, support collection, and carry out the necessary communication regarding the situation with the community, authorities, blood donors, and other actors, important actors. In this slide, we can see the voluntary blood donation and reducing voluntary blood donation and the reduction of the risk of TTIs are directly related to the efficiency of blood bank. This is the situation in PAHO region. Banks with greater capacity for blood collection and processing generally have more personnel dedicated to promotion and collection, and they also implement good manufacturing practice, which leads to improving the availability and access and safety of blood and a more effective cost for the health system. For this region, wherever possible, to develop strategies for centralization of processes or banks, it should be considered to improve the efficiency of the system. In accordance with PAHO and WHO recommendation to maintain a safe blood supply in the context of the COVID-19 pandemic, the countries assess blood need, verify blood inventories, and in some cases move the blood to areas with risks of viral infection and low blood coagulation. They evaluate need of blood versus blood availability before starting to make call to the community. Voluntary and unpaid donors attending, they make donor mobilization with the health authorities is very important. Donor transportation and pass or ticket to move the blood donors in times of lockdown. And other activities were the countries implement recommendation to blood service about the COVID-19 donation guidelines, prescription of use of blood, monitoring of the inventories, and mobilization of blood or redistribution or exchange in different cases. According to a quick survey in 2020, we saw that the system with the highest collection capacity and the highest percent of voluntary blood donation had the least impact in the blood supply. This is very important for the region. Now we are working analyze and evaluate the impact of the pandemic in blood supply and evaluate the compliance with the contingency plan and evaluate the coverage, blood collection, and distribution and use of the indicator by regions. Continue with the inventory control and management of stock of blood. To inform the competent authorities of the action carried out. Inform and thank the community for the response to donating blood in this emergency. Finally, the region, the Bajo region, has made important advance in safe blood and increased collection capacity. And we can see the coordinated service organization improves efficiency in obtaining and supplying blood and blood component, more units, availability and lower cost, and more capacity, especially with it have extramural collection. It has extramural collection or donor extramural campaign, and increase percentage voluntary donation. Countries with voluntary blood donation, organized service, emerging blood management plan, and coordination and communication mechanisms have a better response to the pandemic. We have a very important information system of the region, and many countries have information system about the collection and distribution they manage. This is a good support to the management in blood emergency. Thank you, many thanks for the attention. Thank you. The floor is for you, Jujun.
Yeah. Thank you, Dr. Beltran, for your presentation that's describing the regional activity in the contingency plan for the blood supply. And I think before we have the discussion session, so we still have two more presentations from our speakers. So the next speaker is Dr. May Rauf. So Dr. Rauf is the director of Dubai Blood Donation Center. I think she will share with us the experience from regional of East Mediterranean and also maybe for Dubai experience on contingency plan during the on blood supply during the emergency situation. Dr. Rolf, please, floor is yours.
Hello, hi everybody. It's my great pleasure and honor, and on behalf of WHO in Eastern Mediterranean region, to have this opportunity to share the experience of the region in this scientific gathering. I would like to apologize on behalf of Dr. Yetim Abdullah, Medical Officer, Blood and Other Products of Human Origin from WHO Regional Office for the Eastern Mediterranean region, for couldn't attend for an urgent matter to present with me. The WHO Eastern Mediterranean region includes 21 member states and Palestine, 583 million population. The region have different country with humanitarian emergencies. The Eastern Mediterranean region for WHO has guided the countries by issuing national contingency plan for blood supply in 2016 and the regional strategic framework for blood safety and availability for the year 2016-2025. I would like to thank the WHO Regional Office in Eastern Mediterranean for giving the opportunity for Dubai in United Arab Emirates to share the experience in contingency planning to ensure adequate blood and blood component supply. United Arab Emirates, UAE, is the federation of 7 emirates and population of around 9.9. The UAE National Blood Transfusion Policy that has been issued by Minister's Cabinet Decree in 2008, it requires related or concerned health authority to ensure that blood donation centers are governmental, they are non-profit organization, promoting and encouraging voluntary non-remunerated blood donation 100%, ensuring that all patients in UAE have an adequate and safe blood supply, and promoting safe and high quality management for blood and components. The need to have contingency and risk mitigation measure in place to ensure continuity of the service and blood supply and blood related product is one of the mandatory for all blood donation centers. And recently, a ministerial decree issued in 2021 requesting blood donation center to start freezing red cell to cover emergency and disasters and also to cover rare blood groups. In UAE, the National Blood Supply Contingency Plan describes the context in which contingency planning is addressed and giving guidance for interaction of contingency management between federal, local health authorities, stakeholders, and supporting services. There is a Supreme National Blood Transfusion Committee, which is chaired by Ministry of Health and Prevention, and it includes in its members from the local health authority and the blood facility in the country. It is responsible for blood supply contingency planning review and approval, and it is responsible for identifying related stakeholders and supportive services and assign roles and responsibility for each one of them. Now, to give you an example of our stakeholders, they include National Emergency Crisis and Disasters Management Authority, Ministry of Health, local health authorities, Armed Forces Medical Services. And for our supporting services, we include Ministry of Interior, local police, civil defense department, local of municipality and transport company, electricity, water authority, and Emirates Telecommunication Corporation. And in our emergency and disaster plan, we have role and responsibility for each one of the previously mentioned. Now, to talk about Dubai Health Authority blood supply contingency planning, the contingency plan is designed to guide the Dubai Blood Donation Center as the only donation center in the Emirate of Dubai and other relevant stakeholders and supporters in coordinating an appropriate local, in case there is any local or national response to the event of domestic threat or disaster that affect the safety and availability of blood in UAE or in Dubai in particular. So for us, we have assigned 3 alerts for the inventory of blood and component, lie under green, amber, and red alerts. And the daily normal communication of blood inventory, it is a live electronic system that communicate Dubai Blood Donation Center with higher management in Dubai Health Authority. And this is also connected to Ministry of Health, which is representing also emergency and disaster committee in the level of the country. So daily life system communicating what is available from blood and component. And I just like to share with you the dashboard that represent how our stock is represented in the operation and command center in Dubai Health Authority. It's showing the available blood stock, which is ready to go. It can show the blood and also the component. And also it is showing that how much blood has been collected, but it is not ready, it is under processing. And in addition to the blood that has been issued during the last 10 minutes, and also it compared the collection versus supply for 24 hours. So the communication structure of a blood inventory in case there is an emergency or disaster, it will start from operation and command center, assigned person called mission controller, to the operation and command center expeditor, to the head of the center, to the director of the department, and the CEO, and then to higher emergency and disaster committee in Dubai Health Authority. So communication in general for blood inventory during emergency and disaster, it will start from the center level to sector level to local level and national level. And all these levels, responsibility and role is well documented in our emergency and disaster plan. So just to show you, like, this is part of our plan, and there is different phases, the green, the amber, and the red phase. And in each one, what is the responsibility and action to be taken by the blood collection center, by the hospital-based blood bank, and how the information is transmitted to Dubai Health Authority Disaster and Emergency Committee, which then, if needed, it will be connected to a higher emergency and disaster committee in the country. Dubai mitigation and the preparedness plan is very well structured. We have a very good business continuity plan. We also have a risk assessment that is reviewed every 6 months. And we include the different scenarios and assumption of a blood supply disaster, including different scenarios. And the most important that we concentrate a lot on employee education, training, and conducting drills. So we ensure that the staff are aware to act properly in case of different scenarios for different emergencies and disasters. I'd just like to share with you this slide that's showing our action that we have taken during and business continuity during COVID-19, during the lockdown. So we have a very good experience of having a very good database of donors who are willing to donate during emergency, as we have released and launched in 2017 a smart mobile app called Dami in Arabic, and it means my blood. And this encouraged people to register themselves in order to donate blood during emergency and disaster and rare blood group. So lots of people were registering, and really we use this information to contact the donor, ask them to donate blood during COVID-19. We also have initiated an alternative collection site for blood, as during COVID, all our mobile buses have been stopped because of movement restriction. So we have structured a very big tent to welcome the donor and to be able to implement social distancing and all the safety precautions to make them assured and make them happy to come and donate during the pandemic. Also, we have issued in 3 languages the reviewed donor eligibility criteria that has been changed a little bit during the COVID-19. And also on the other side, we have monitored daily the donors recruitment tool. So we were sending SMS to the donor, asking them to donate. We're sending emails, making direct telephone calls. And also through social media, we were announcing and posting call for donors to come and donate. So we were monitoring daily, like what is the strongest tool to make the donor come and donate. And really, it has been changed during the COVID pandemic period. Like, and sometimes it was the social media, and another time is the SMS, and so it was very helpful for us to monitor these operational aspects. During COVID, early in the pandemic, we have announced yellow alert to hospitals to prioritize their patients, and this is also available in our emergency and disaster plan. We are very well identifying the priorities of patients in case of disaster. We have also postponed all cold surgeries in the Emirate, in the government and private sector. So we were so dependent on social media activity in announcing the working hours that has been extended and changed, and also like encouraging and assuring the public about the safety of their donation during the pandemic. Also, the center, beginning of April 2020, we start collecting convalescent plasma to help in treating patients with COVID. As a conclusion and to summarize the lesson learned from the pandemic, it's very important to have a structured business continuity procedure in addition to emergency and disaster plan, and get ready and prepared for implementation. Collaboration and communication between blood donation center, higher management, emergency and disaster committee, our stakeholders and supporting services is really very important, and we need to have drills at least once in the year to be ready for such communication. Importance of having donors database and encouraging public to register for donation during emergency. We found it really very useful, as the donors who have registered and we contacted them, most of them, they were able to come and support us. The availability of enough stock from consumables, reagent, kits, which we call it a strategic stock, is very important and essential. And in UAE, as per Minister's Cabinet decree, we should have a strategic stock for at least 3 months available nearby that we can access in case there is any, like what happened, lockdown, and we cannot receive items or reagent from outside the country. Media support is very important to ensure that public safety during donation and encouraging people to donate. In my last slide, I would like to share with you that United Arab Emirates in 2008 has hosted the global celebration of World Blood Donor Day as the first Arabic country, and it was under the theme of many happy returns. It reflects the support of the higher management in UAE for voluntary non-remunerated blood donation. Thank you so much for your attention. Thank you.
Thank you, Dr. Ralph, for your presentation. It seems that the National Blood Supply Contingency Plan in Dubai is very well established and well functioned, so congratulations. Thank you. Now we can move to our last speaker, Dr. Emmanuel Capobianco. Dr. Capobianco is the Director of Health and Care from the International Federation of Red Cross and Red Crescent. I think Dr. Capobianco will share with us the role of the IFRC in achieving the universal blood supply. Dr. Capobianco, please, floor is yours.
Thank you very much, and warm greetings to you all from Geneva. It's a great pleasure to be with you today, and I look forward to share a bit of the experience of the International Federation of the Red Cross, Red Crescent, that brings together 192 national societies, Red Cross, Red Crescent societies that will operate all over the world. I will focus on three main points. The first one being the impact of disasters on blood and the importance and the critical priorities for the response.
Dottor
The second,
Capobianco,
following...
se può modificare display setting, ce lo dà in...
We kindly ask you to please change your display settings, please.
La ringrazio, scusatemi. The second point would be the business continuity planning, as mentioned already by Dr. Raouf and other presenters before her. And last but not least, I will focus on the blood program recovery. Let me start maybe from something that is very simple, but the fact that disasters, being hurricanes or earthquakes or a tsunami, they increase trauma. And so during a disaster, there is a peak demand for blood. But what we see in disaster is that the provision of blood is severely impaired by a number of factors. First, damaging in the infrastructures and the equipment. Second, problems in access to blood donors. And we have seen this very clearly over the past several months with COVID-19. We have issues of the supply chain that may be interrupted, causing shortages of blood bags or test kits, for example. And we do have disrupted transport network with restricted ability to move blood around the city or even in larger geographies. Now, we in the Red Cross, Red Crescent movement operate full blood programs in a number of countries. In some countries, we complement government supplies, but in many countries, the Red Cross, Red Crescent is the only supplier of blood. And when a disaster happens, we know that the demands will increase, and there are two main priorities. One, to rapidly restore the blood supplies, and second, to immediately start with the rebuilding and the recovery of the blood program for the medium-long term. And we know that this risk is very much mitigated through what was, again, presented by the previous speakers, the need of a business continuity plan. And I will be focusing on the critical elements of the business continuity plan from our side. We know is essential to manage our response so that the staff, volunteers, and partners are very clear on what should be done, by whom, and when. It's critical for the response, but it's critical for the mitigation, the preparedness, and the recovery. One critical point for us is the fact that the plans for blood needs to be well integrated into national disaster plans. And to do so is very important that the blood donation is recognized by the government as an essential service. So the three elements of business continuity plans that we wanted to highlight here are related first to management, having a clear management team with well-identified roles and responsibilities about the members of the team, clear contact persons and stakeholders, having all donor records in clear arrangements for the storage, and computer backup system in where in use. So management is the first element, people is the first element, but communication is just as important, and it's a communication that goes internally, that needs to bring staff and volunteers all together, but also needs to go and be oriented towards the outside, including government, donors, media, hospital stakeholders, and other suppliers. The third element, just as critical, is the infrastructure, and ensuring that the plan includes alternative sites for blood collection, processing, and storage in case of an earthquake destroying the primary site. We need to have contingencies for blood transport and remote storage. We need to have alternative suppliers of equipment and consumables, and also identify procedures for blood collection and processing during a disaster. Of course, it's very critical to design or modify permanent facilities to withstand disaster impact. As we saw in the previous presentation, business continuity planning is not a static exercise. It's an exercise that needs to be reviewed on a regular basis to ensure that the plan remains up to date, and understanding what challenges may be happening in terms of diseases or events, but also taking into account the changes that may happen over time to blood program processes. The business continuity plans needs to be tested so that staff can know exactly what to do and act immediately. And then they need to be evaluated when a disaster strikes so that the learnings can be incorporated and all the actions and outcomes can be recorded. As I said at the beginning, the response in immediate aftermath of a disaster is of paramount importance to save lives. But building on the recovery of blood programs within the Red Cross is a top priority that initiates immediately when a disaster strikes to ensure that we include in the emergency appeal that we launch to collect funding to respond to a natural disaster. We ensure that blood is immediately included, and we do have assessments that are conducted immediately after the disaster by the teams on the ground to identify recovery requirements across all critical areas of the blood program. And then we work as a Red Cross in collaboration. We are a movement, as I said, of 192 national societies, more than 14 million volunteers. We do have a global advisory panel based in Australia that is the entity that technically support national societies providing assistance in the recovery phase and linking with the overall response that the IFRC is setting up either from a regional basis or from here in Geneva. And what we strive in the recovery is not only to bring back the functionality of a blood system in a country that may have been damaged, but use the opportunity within brackets of a disaster to strengthen the capacity to build partnership around the blood programs that can ideally build the program better. And with this, I will stop and give you back the floor. Thank you very much again for the opportunity of being with you.
Thank you very much, Dr. Capobianco, for your presentation. So I really agree with you that blood should be included in any disasters appeal. And now I think we can have the question and answer session. So I will hand over this floor to Dr. Giangelis to lead the discussion.
Yeah, thank you very much. Thank you really very much for the presentations. They are very helpful in letting us to imagine how we can look at any possibility to have a contingency plan on blood supply, which is not only blood by itself and donors, but also everything is needed to have the blood components and the plasma-derived medicinal products prepared in order to be given to the patients. There is something that I remember we are discussing now in this period, is that there are also some long-term effects of the shortage due to the pandemic, and this long-term effect is the probability, the possibility that due to decrease in blood donation and in plasma collection, then we will face some decrease in availability of medicinals as immunoglobulins, as albumin in the next future, because you know, there is the lag phase between the collection of plasma and the production of medicinals. And so if as it happened during the pandemic, we have a decrease in collection of plasma, then we will expect a shortage in the production of medicinals after a few months. But I have two questions in my chat, and if someone wanted to ask something to the presenters, please use the chat and put the questions in the chat. So I will begin with two questions coming both from my co-chairperson. The first question to Dr. Rauf is, after thanking, of course, for sharing your experience, how to integrate the blood supply contingency plan into a national emergency contingency plan, which is something which has been recalled also by Dr. Capobianco. Well, who, in your opinion, or in the opinion of other speakers, hold the key role for this successful integration?
In United Arab Emirates, we have 4 main blood donation centers, all belong to the government. And we have a higher emergency and disaster committee. They have a subcommittee, which is just for blood supply. So we decided before 2 years as members in this committee to have a national blood emergency and disaster committee that connect all the blood donation center. We agreed on having our supportive and supporting institutes and also like our stakeholders. Concerning the daily connection that the stock is available for all higher authority and key people, it has been done 2 years back by business intelligence system. It is an IT network that connects the different systems available in each blood bank, though in each blood donation center, though our blood donation center having different information system, so different systems, but they can extract certain data related to the available stock, and it can put it in a system or in a dashboard that can be observed and governed by a higher authority. So in case there is any shortage or any emergency in one of the blood donation center, then there is a very well structured plan for the immediate action to be taken by the geographical area nearby. And in case this is not enough, though, it will be cascaded to higher community. So we started with an emergency and disaster plan, and we are conducting the drills. And the main part of this plan is to have a network between these donation center and the stocks availability, and the threshold for each center is different from the other, but we are connected to one system for governance. I hope I answered the question.
Thank you. And from the speakers, also Dr. Capobianco asked to put a comment on this question. So please, Dr. Capobianco.
Thank you. Very briefly, just to say that disaster management very often sits in civil protection systems, and you do have a public health emergency response that often sits outside in Ministry of Health. And I think we saw sometimes the lack of coordination between the two, particularly during the COVID-19 response. I believe, and we at the Red Cross believe, that moving forward after this painful experience of COVID-19, it will be truly important to bring these two elements much more aligned, having the Ministry of Health and the plans within health much more aligned with the Ministry with disaster management. And right now there are some hybrid systems, but they are not. This is not generally what we find, and I think this will require intervention at the low level. We really need to bring in our disaster law and ensuring that the elements of public health are very much included, so that there is a framework on which we can operate in the future. That's a part of the work that I think we all have to do from a blood perspective, but mostly from a public health perspective, to link public health emergency to disaster response. Thank you.
Thank you, Dr. Capobianco. I have on the chat another question. The question is, so far, WHO has developed the guidance on managing blood supply during disease outbreak. And the question to Dr. Shah is, do you think there is a need to develop the guidance on managing blood supply during natural disaster or other kind of disasters? And let me add to this question just another question. And do we need to think not only to reduce the availability of blood because any kind of natural disaster, but as occurred during this pandemic, it was a virus not spread through the blood, but it was an airborne threat to all the donors and the staff during their job in donation and in collection of blood. So it is a matter of safety of the environment where you and the donors and the staff are working. So it's really a huge amount of number of disasters that you have to look at in order to define a precise contingency plan on the availability of blood components and plasma for patients. So.
Thank you. I guess this question is from Yuyun. So, very good question indeed. And I would say this topic is covered in bits and pieces in emergency management guidance documents of WHO at several places, but I think it would be a good idea to consider to cover this topic, and especially at the time when we are discussing our technical products for the upcoming biennium, it would be a good idea to give a, we can think about developing this. And Chair actually said, and I will agree that COVID-19 is a very, very unique scenario, though it is not transmitting through blood, but it is still kind of causing problems in blood collection. So in a way, the guidelines which we are giving for COVID, to some percentage, it is also contributing to teaching member states how to overcome blood shortages in even natural disasters, going to the places, going to the donor's place, using mobile vans, collecting blood, seeing which of the part of country is not affected, kind of things. So, but still, it is in pieces. But if we wish to give a solid answer, we need to have separate guidelines. And I'm sure Yuyun will definitely consider this in our upcoming technical products, and we can definitely work on this with our all relevant stakeholders. Over and thanks.
Thank you. And I have in the chat Dr. Mauricio Beltrán Durán, who is asking to take the floor for some comments on both questions. And then I have a question from Miguel Lozano. But first of all, please, Dr. Durán.
Thank you. Thank you, Dr. Vincenzo. In the PAHO region, especially in the Caribbean countries, we have frequently hurricane. And for this case, the PAHO produce guidelines about how avoiding blood emergency in this type of disaster. In this case, many countries in Caribbean and Latin America, we have a national blood coordinators or national blood directors program, and they coordinate with the global authority that are responsible of emergencies in each country. In this case, we support with these guidelines, and we support with the national blood program coordinator to include in global emergency or national emergency group. In general, in Latin American countries, especially, the national blood coordinator are included in the committee of response, global response in each country. Nance.
Thanks. I have now two questions, one by Dr. Michele Lozano to Dr. Capobianco. The question is how easy or difficult would be to send blood components to another country struck by a disaster? And the second question comes by Dr. Montrasio, who asked if the people can share the experience of managing shortages and principles chosen for priority allocation during COVID-19 crisis. Thank you.
Shall I go first?
Yep, of course.
Mr. Chair, yes. Just say it's fairly complex to transfer blood components. So the best approach is really to establish capacity at the local level and re-establish the supplies at the local level. That's how we come in and try really to support the affected regions. Usually what you do have in natural disasters is that they are localized. There is a capacity within a country that can surge from regions that have not been affected by a hurricane or an earthquake that may affect just part of a region in a serious way to impact the blood services.
And there was another question to the speakers related to, do you have any experience, how did you define, or how, if there are plans to define, in case of shortage, short-term, long-term, middle-term, long-term shortage, which are the principles you can use for priority allocation of resources if they are decreasing the availability? I think I try to give my opinion on that. I think that the most important thing to do is to define a sort of list of conditions in which you cannot postpone the use of the resource of the limited resources because it is really life-threatening. So you have to define the situation in which you are not allowed to postpone the supply of the resources from conditions in which you can wait for some moments sometimes. And I think that it is really a matter of scientific evidence on what is really needed and what is under scrutiny, let me say, in some way. Yeah, please.
Thank you. This is a very important aspect to include the coordinator of blood or the National Blood Service in the committee of the emergency, because he have the access to the epidemiological information. And in this case, he can review where is the some of the areas that have many problem or more problem about the circulation of virus or the event or the different event. It is important when the coordination or the director of blood participate in committee. The other aspect is in the national plans or emergency plans, it's very important that the countries have different alternatives, the 2 or 3 or 5 big block ban in different allocation, in different geographical allocation, especially when the country is big, or 1 or 2 alternatives. We can have. We can move, in this case, the blood units in these territories when you have a backup, or 1 or 2 blood bank in backup. This aspect need include in the national blood emergency plan. Thank you.
You? Anyone else who want to take the floor? And
Yeah, I saw Dr. Rolf hands up. Yeah.
Yeah, thank you. So I think that an emergency and response plan for any donation center, they should include, according to the geographical area that is covered by this center, the threshold or the phases, like the green phase when the normal situation, or yellow where we are going to the red zone. Of course, our target is never to reach the red zone or the red alert for blood supply. And the most important is when you prioritize the clinical condition in the plan, it needs to be communicated with the clinicians, because in many cases, clinicians are on one side and the blood centers on other side. So we need to have a good communication of this plan with the clinicians for them to understand and to support the blood services while prioritizing the conditions to use the blood, and for them to know the situation continuously, so they will be prepared and ready to support us. Thank you.
Yeah, I fully agree with you, and it is a matter of clinical decision. And so the agreement between the blood establishment and the clinical side is essential to define this priority list when we have a shortage. If there are no other questions or comments, we are on time. And I would thank you all, thank really all of you, all the presenters, for the very, very nice presentation. The discussion was incredible. I was thinking that probably we were going to end the session in advance, but it didn't happen because the discussion was really very, very important. And so I would thank really very much all the presenters and my co-chairperson for conducting this very important session. So now we have 15 minutes for a virtual coffee break. And so we will take advantage of this virtual coffee break. And we'll see here at 14:45 time of Rome, of course. So in 15 minutes, again back for the third session. So see you later.
Thank you.
Thank you, all the speakers. Thank you, Dr.
Bye.
is centered on the country perspective and initiatives on donor management. And let me introduce the chairpersons of this session, Dr. Karin Magnussen and Dr. Jumpin Yu. First of all, Dr. Karin Magnussen, she is the medical director of the Department of Blood Center Medical Biochemistry in Glandet Hospital Trust in Norway. She is well known being the chair of the International Federation of Blood Donor Organization Medical Counselors Group, and she is also the medical advisor of the Danish Blood Donor Association, chair of the ISBT Working Party of Donor and Donations, member of the American Association of Blood Banks Committee on Donor Health and Safety, and representative Union of European Medical Specialists, Sectional Laboratory Medicine, Chair of Division of Hematology and Fellow of the European Board of Laboratory Medicine. She has 45 publications on peer-reviewed indexed medical journals in the field of transfusion medicine, anemia, hemochromatosis, donor hemoglobin and ferritin, and donor healthcare. So he's the right person to be one of the chairpersons of this session. And as I can personally confirm, she is a very good skier. And Dr. Jumpin Yu, who is the leader of the technical officer of the WHO Department of Blood Safety. So welcome to both the chairperson, and the floor is yours.
Thank you, Vincenzo. You are a much better skier than I am. Welcome, everybody, to this interesting session. I will try to share my screen. Here we go. So I'm very delighted to welcome you all to this session on donor protection. I'm sure it will be very interesting. I hope you are well rested after a few cups of coffee. Donors, as you all know, are the most important link in the chain to be able to treat patients with life-saving blood products. And we will hear presentations from three excellent speakers with Steven Stidnalnik from Columbia University Center in New York on From Donor Biology to Donor Health Protection, and Dana Devine from the Canadian Blood Services on the present of transfusion medicine from donor gift to the best transfusion product. And last but not least, from Eldad Hot, also from Columbia University Center in New York on donor recipient and ion alliance. I'm looking very much forward to hear these presentations. Welcome to the speakers, and first you, Stephen, thank you.
Can you stop sharing your screen, Karin? Thank you. Okay. Thank you, Karin. Thank you very much to the organizers for inviting me to speak today. These are my potential conflicts of interest, and I don't think any of them are relevant to today's talk. And this is the group of people I have the honor to work with every day at Columbia University. So one of the central thoughts of our research is to see if we can treat blood products as pharmaceuticals. That is, can we think of complex biological products as pure drugs? As an example, if you get a headache as a result of my talk, you could brew yourself some white willow bark tea, and that will make your headache go away. But the medical model is to think about what the active ingredient is in the white willow bark tea, see if it can be purified and then dosed appropriately and dosed specifically. And of course, aspirin is the active ingredient in white willow bark tea. So there's actually a medical model of a pharmaceutical, which contains these kinds of characteristics that should make sense to you, that what are the active ingredients, how pure is it, what dosage should one use, is it clinically effective, what are the indications, et cetera, et cetera. And we think that you can apply all of these types of characteristics when one is thinking about blood products. So the idea is, can you turn complex biological products into pure drugs? And this might sound fanciful, but actually has been done. And so when I was a resident, we would treat hemophilia A patients with cryoprecipitate 45 years ago, and now we use recombinant factor VIII. Of course, it's a little bit more difficult to construct a red blood cell in this way, but various groups are studying what's called blood farming, and maybe in my lifetime or your lifetime, we will see this in practice. However, until we get there, one of our approaches is, can we think about making better starting material? And again, if we go back to white willow bark tea, it's made from the bark of the white willow tree. And so you could imagine if you wanted to make a higher active such therapeutic, that you might see if there are subspecies of this tree that have higher amounts of salicylic acid in them, or are disease resistant, or grow faster, or produce better bark, or something like that. And you can think about the same kind of concepts when you think about red blood cell units, which is the focus of my talk. And here's an example where even the amount of hemoglobin in a unit of blood at collection varies tremendously, from units that are fairly low to units that are fairly high. And so the dose of hemoglobin that we're actually giving to a patient will vary tremendously based on what the hemoglobin content of that unit of red blood cells is. So can we think about this, and can we think about better ways of collecting, processing, and storing blood? And again, if we go back to the tree, here's the tree, and we need to take the bark off the tree. Are there better ways of removing the bark? And are there better ways of purifying the active ingredient to make better drug that's cheaper, that's more standardized, et cetera, et cetera? So in the context of red blood cells, what happens to a red blood cell as we store it in the refrigerator? Well, this is actually a fairly complicated process called the red blood cell storage lesion. I'm not going to go into it other than showing you two pictures. So this is a picture of what red blood cells look like, scanning electron micrograph, when they're collected fresh, beautiful biconcave discs. But at day 42, at outdate in the United States, this is what those red blood cells look like. They're smaller, they're rounder, they have spikes, and these are in fact the red blood cells that we transfuse into patients. And in Europe and the United States, at the least, there are quality criteria on how we should store red blood cells. And so as storage time increases, we want to minimize the amount of hemolysis in the storage bag, the amount of red blood cells that explode and release hemoglobin. But in fact, it's virtually never zero. So we're always infusing some free hemoglobin and other red cell stuff into patients. Similarly, a second criteria is that the red cells should still circulate post-transfusion. And in the United States and Europe, the goal is to have at least 75% of those red cells circulating 24 hours after transfusion as the storage time goes on. But obviously, at the end of storage time, there will be less of a hemoglobin bump in the patient than at the beginning of the storage time. And so we're giving different doses of this drug to patients depending on how long they're stored in the refrigerator. And so we may be giving less than an optimal dose. And interestingly, one of the things we should be worrying about is, do the transfused red cells actually have a therapeutic effect? And it is curious that there is no such quality criterion to demonstrate or prove that the red cells that we transfuse have a therapeutic effect. So what happens in humans, actually? And this is a summary of over 600 such post-transfusion recovery studies that were performed. And as you can see, they're pretty good. So most of them are greater than 75% of the red cells circulating 24 hours after transfusion. However, there are some donors where the post-transfusion recovery is actually pretty low. And then there are some donors where the survival of these transfused red cells, even after 6 weeks in the refrigerator, is remarkably high. And we understand relatively little about what differentiates one donor from another. And so I would like to just give you 3 tiny vignettes about different kinds of characteristics that we can think about in terms of the donor that affects the quality of the red cells that get transfused: genetics, diet, and environment. So in the context of genetics, one can ask the question of whether there are any specific genes that are involved in red cell storage quality. And we in our laboratory are particularly interested in a gene called G6PD, or glucose 6-phosphate dehydrogenase, and in donors who may have G6PD deficiency. There are about 400 million human beings around the world who have G6PD deficiency, and this is a genetically induced variation in the enzyme activity, which confers resistance to oxidative stress in red blood cells and other tissues. And one could ask the question of whether this confers resistance or susceptibility to the red blood cell storage lesion. And in Rome, which I wish I were right now, the major inciting event is fava beans, and these are lovely Italian fava beans that could induce a hemolytic crisis in a patient with G6PD deficiency. And so we did a study comparing donors who were G6PD sufficient or G6PD deficient. And you can see that the post-transfusion recovery in the G6PD deficient units was less than in the G6PD normal units. And in fact, 3 of these individuals had red blood cells that stored beneath the quality criterion that is set by Europe and the United States FDA, so that these red cells store less well than red cells from G6PD normal donors. And so it raises the question of whether we should be screening all of our donors for G6PD deficiency, particularly in a place like Italy, where G6PD deficiency is fairly common. And if the units are G6PD deficient, should we store them differently? How does this affect the quality of the red cells that are transfused into recipients? And ought we to provide medical advice to donors who turn out to be G6PD deficient and advise them, for example, to avoid eating fava beans? The second vignette concerns diet. And there are many things in the diet that affect the ability to produce a robust red blood cell. And one of these is iron. And we know that iron deficiency is very common in blood donors. And another speaker, my colleague, Dr. Hode, will be talking about this in his talk shortly. But it also raises the question of, should we be screening all blood donors for iron deficiency? If the blood donor is iron deficient, should we store their red blood cells differently? How does this affect the response, therapeutic response in recipients? And also, should we provide advice to these donors as to whether they should treat their iron deficiency and how best to do that? And then finally, what I'm calling environment, that other people are now calling the exposome. And that is that human blood donors are exposed to all sorts of things. Many of them, like me, are getting older. Some donors smoke. The gut microbiome varies. The donor can be on various therapeutic or recreational drugs, shall we say. And one component that I'd just like to mention is lead. And lead is an environmental pollutant that still is remarkably present in the United States, for example. It has toxic effects on brain development. There's thought to be no threshold effect, so any exposure to lead is potentially damaging. Premature infants whose brains are growing and developing are particularly vulnerable. And in a blood product, if there's any lead, almost all of it is in the red blood cells. And so we know, actually, that some donor red blood cell units contain high lead levels, and that this lead is actually transferred to the recipient by transfusion. And there's a wonderful study published a few years ago from a Canadian group about this topic, in case you're interested. And so again, it raises the question, should we screen donors? Should we store them differently to protect recipients? Should we not transfuse units that have lead in them to premature infants? And should we also provide medical advice to the donors on how they can reduce the toxicity of lead if they happen to have lead in their blood? And so the goal, I think, of transfusion medicine is to make an ideal red blood cell unit. And if we did that, it would be that the post-transfusion recovery and the lifespan would be equivalent to fresh blood. Ideally, there would be no white blood cells so that we could avoid certain kinds of transfusion reactions. There'd be no plasma so we could avoid other kinds of transfusion reactions. They would lack clinically significant blood group antigens so we could avoid hemolytic transfusion reactions like ABO. And ideally, obviously, that there would be no transfusion-transmitted infections. So how are we doing in 2021? Well, actually, we're not doing too badly. So over the last 20 years, there have been novel storage solutions that have been developed, and we're developing ways of selecting and preparing blood donors better. For white blood cells, we can leukoreduce those units, we can irradiate the units. For plasma, it's possible to wash the units or select the right donors like in IgA deficiency. And there are many groups that are using genetic matching between donor and recipient, particularly for patients who are chronic transfusion recipients. And then finally, for transfusion-transmitted infections, there are new screening tests every year. Now pathogen reduction and activation is available in certain settings. And of course, on World Blood Donor Day, what would be better than celebrating Harvey Alter's recent Nobel Prize for hepatitis C? So in my opinion, in terms of protecting our donors, I think it would be very important to understand donor genetics, understand donor diet, and understand the donor exposome. And of course, with the consent of the donor, we could use this information to maintain and improve donor health, which I think would be highly desirable. And so finally, I would like to thank, again, the organizers for inviting me, and in particular, thanking all of the altruistic blood donors around the world who make what we do possible. So now it is my pleasure to turn the podium over to my good friend from Canada, Dana Devine. Thank you very much.
Thank you very much, Steve. And just let me share my screen here, which hopefully everyone will be able to see at this point. Okay, so thank you very much for the invitation to speak at this meeting. I'm really thrilled to be here, and just a little bit sorry that I'm not actually physically present in Rome, which is probably my favorite city in Europe. I was asked to speak a little bit about some of the things that we're starting to understand about the relationship between the blood donor and the quality of the transfusion product. And so that's what I'd like to do. I have a few disclosures. I don't think they have any relevance to what I'm talking about today. So the real question that we have is as Steve was indicating, is really what happens to blood cells in storage? And as he mentioned to you, all of the cellular blood products that we put into plastic bags develop storage lesions over time, and that most of these actually have a negative impact on blood quality. You've already seen this graph, but the reason that I put it in here into my talk was to show you that if you just simply take survival and recovery studies, and these are from normal volunteers, that you do find people whose red cells don't store very well and the recoveries are very poor. And so there's a collection of folks here who are all underneath this red line, which is the FDA standard for survival and recovery if you were, for example, going to introduce a new blood bag to the market. So these folks provide some interesting study material because it would be very good to be able to understand why their red cells are not storing well. This is data from a very interesting paper that was published in Blood last month, which was actually looking at several things. One of which is, as what Steve was describing to you, is the marked morphologic change in red cells as they're put through storage. And as red cells are kept in a bag, they start to spit off little bits of membrane that we call microvesicles. And what's left behind is actually a smaller erythrocyte. So these are what are called storage-induced microerythrocytes. And you can see here that if you count them up in various blood donors, that they're very different. So you have some donors who are producing very high levels of these storage-induced microerythrocytes, which are very rapidly cleared on infusion, and then other donors who don't produce as many of them. So trying to understand what actually puts a donor in a high producer versus a low producer would be very important to help ensure the quality of blood products. So what we're trying to do in my research group is really understand what factors are known to contribute to blood product quality. And there's data in the literature that's really quite interesting. So this graph on the left is from the REDS-3 study, which very clearly shows that there's a significant difference between males and females with respect to storage hemolysis, and that at least for males, this starts to widen significantly in the last decades of your donating life. At the same time, some of our colleagues in Greece have done some very interesting work looking at the difference between osmotic fragility between women and men, and also in the total antioxidant capacity, which is what's shown on this top graph. Similarly, the body mass index or the triglyceride levels also matter for how hemolyzed you are. Again, on the left-hand side, these are data from REDS-3 showing in the black triangles that the higher your body mass index, the more hemolysis you get during storage, and that this, at least in the latter weeks of storage, this is significantly different from people who have lower body mass index. My group has looked at the triglyceride levels in people that we've identified through the Canadian Blood Services Quality Control Program as having high hemolysis at outdate. And at least in our male donors, we're able to see that the triglyceride levels, if they're high, are higher in the high hemolyzers than they are in our controls. This is a very small sample size on female high hemolyzers, and so I don't want to make too many conclusions from that. So red cells aren't the only product that age in a plastic bag. Platelets do as well. This is a very interesting set of quality control data that were pooled through the BEST collaborative across a series of blood operators. So represented here is one of the large blood operators in the US, the two Canadian operators, the Dutch, the French, and the NHSBT in the UK. And what we all did was we went into our quality control databases and we asked the question, if you had a donor who showed up a first time in your quality control data, do they show up a second time? And if they did that, we took both of those values and then plotted them. And what you can see in this red box is that donors who actually make a platelet apheresis product that fails pH at outdate is highly likely to fail on the second time. And so this is actually a very good way to capture donors whose products are not going to store well. And so we need to have more ability to actually look at these things, to identify biomarkers that can pick these donors out of the donor pool, because we know that if we're giving platelets that have a pH of 5.7 to recipients, the platelets don't stay in the circulation. They're essentially immediately cleared. So those would be good products for, at least from the blood operator perspective, for us not to put on the shelves. So my lab has taken us another look at this by actually looking at healthy volunteers, and we did this in collaboration with Cheryl Slichter and the group in Seattle, who do a lot of radio-labeled survival and recovery studies for platelets in normal volunteers. And we asked them to give us some samples from donors whose platelets stored well and other donors whose platelets did not store well. And what we did was subject those to a proteomics analysis. And we identified two signal transduction molecules, RAP1 and Rho-GDI, that are present in platelets that actually show a very strong correlation with the recovery and survival of those platelets when they're infused back into the person that they came from after those platelets have been stored and radio-labeled. So this started to make us think it might be possible to identify biomarkers. So the thinking around the biomarkers would be to have all of these happy donors that come to see us, for whom we're very grateful, and that we screen those donors using a slightly different set of criteria. So we expect to find some donors who are not going to store very well, and we'll ask them to contribute to the blood system in a different way. But we will also be able to identify donors who we're quite comfortable are going to have a good outcome. If we're able to do this, we should actually be able to improve the overall quality of the platelet products that we put on hospital shelves. So the questions become, how do we optimize the quality of a donor's gift? Well, we want to be able to ensure that component production and storage are optimized. And we have still quite a bit of work to do on improving storage containers. Obviously, there's a lot of concern going on right now related to the kinds of plasticizers that we're using in storage containers. But there's also a biocompatibility question about our storage containers. We need to have better storage solutions. These are all live cells that we're storing, and they're metabolically active, and they're busy consuming all of the energy sources in these products as we store them on, for example, for platelets at room temperature and shaking incubators. We also need to understand this contribution of donor variation to the product quality, and that requires us to do some more research that's going to help us understand how the storage lesion develops and what the biochemistry of that is, so that we know that well enough to be able to identify these biomarkers that will then help us as blood operators guide how we manage blood donors. So that someday in the future, we might be in a world that looks like this. So if we have biomarkers that can help manage donations, we might be able to say to a donor, Mr. Smith, your biomarker profile is telling us that your platelets aren't going to store very well, so would you please donate whole blood or plasma today? And so this would give us an ability to improve the quality overall of the products that we issue for patients, and that we would also be able to ensure that the quality of the donor's gift is optimized. So in conclusion, the quality of blood products is very important for our patients. We need to work to improve our approach to quality assurance for blood components. And this effort has to include the development of better standards for component quality control and the identification of donors who have characteristics that cause reduced quality. So thank you very much for your attention, and thank you again for the invitation to participate in this meeting. So I'm going to stop sharing and would like to hand the baton over to Eldad Hode. Eldad.
Thank you. Let me share. Can you see my screen? Thank you. Thank you, Dana, for introducing me, and thanks for the organizers for giving me the opportunity to speak today. And like Dana, I really wish I was in Rome giving this talk. So I unfortunately have no conflicts of interest to disclose. So I study transfusion medicine from the perspective of iron biology. And so when I look at a donor donating blood, and this is a little bit oversimplistic, but I view red cells as sort of a bag of iron. And so in addition to just donating blood, they're also donating a tremendous amount of iron with that donation. And the consequence of that is also that when you transfuse a unit of blood, you're also giving a lot of iron to the recipient. And sometimes in certain situations, that load of iron can be too heavy, as depicted in the cartoon. And I've spent most of my career studying the recipient side, but we've also started to examine what happens to the donor when they donate too many units of blood. And so I'll try to share with you three relatively brief vignettes. The first two will have to do with the recipient and what happens when you give a recipient too much iron acutely, and then what happens more chronically when they get too many red cell transfusions. And then finally, I'll end up with the donor side and what happens when you have too little iron. And so as we've seen from the prior two speakers who showed similar images, one of the consequences of putting a bag of blood in the refrigerator is that there's a storage lesion. And just by looking at them, you can see that some of the red cells are damaged after 40 days of storage in the refrigerator. Or the longer you store them, the more damaged they get. And because of time, I won't show you all the data, but we do have data to support what I'm going to show you in these cartoons is that what ends up happening is the longer you store a unit in the refrigerator, the more red cells are delivered to the monocyte macrophage system after transfusion in the recipient. And so you give a fresh unit, most of those red cells will circulate very well. But with an older unit of blood, many of those storage damaged red cells will get cleared from the circulation. And when hemoglobin gets processed, it gets converted to bilirubin, and also releases a tremendous amount of iron. And that iron can leak into the circulation, thereby saturating transferrin and then producing a form of iron called non-transferrin bound iron. And I'll just show you just briefly, why is having so much iron after transfusion, at least acutely, potentially bad, and I'll show you what happens in animal models with infectious risk. And so in the lab, we've developed models of blood banking for mice. And so what you're seeing is a unit of blood that we generate, leukoreduce, manufacture as similar as possible to a unit of blood for humans, and then store it in the refrigerator. And so we can transfuse that unit either fresh or after sitting in the refrigerator for some time so that the red cells get storage damaged. And what you're seeing here is one experiment where we gave 5 mice in each group either a fresh red cell transfusion or an older red cell transfusion that are storage damaged. In addition, we gave these mice bacterial peritonitis with E. coli. So they each got 1 million CFU of E. coli into the peritoneum, and the E. coli expressed luciferase, so they produced light. And so what you're seeing there is the color is the peritonitis that we induced 10 minutes after injecting the bacteria. By 4 hours after injecting the bacteria, you can see that the peritonitis has spread and all the animals are now infected. But by 48 hours after transfusion, in the mice that got the fresh transfusion, they've all been able to clear the infection for the most part, and they're all running around the cage. But the ones who got old blood, you can see the X's means that we had to euthanize them because they didn't look well. And you can see that these mice are completely septic, and so they weren't able to control the infection. This is more complicated. This doesn't work the same with every type of bacteria, and once you throw antibiotics into the mix, it complicates matters more. But this is one of the things that might occur if you give people iron acutely. And so just as an interim summary, our data suggests that red cells transfusion cause hemolysis and acute increases in iron, transferrin saturation, and non-transferrin bound iron. Are they harmful? So I can say for sure in mice and dogs, because there have been some studies in dogs as well, that in certain infectious circumstances, this can be harmful. In humans, this is controversial, and I would argue that it remains to be determined fully in humans whether this is a problem or not. And so I showed you what happens to an infection acutely, but what about too much iron chronically? And so we know that if there are many patients that require chronic transfusions and that this leads to iron overloads, which is one of the consequences of too many transfusions. And so we began looking at how chronic iron overload affects the gut microbiota, or the microbiome, as it's known, and hypothesized that when you have more iron, it can shift the microbiota to more pathogenic species. And so, again, we typically start our studies in mice before moving on to humans, so I'll just show you mouse data. But in these experiments, we have mouse cohorts that are just put on different diets. So the diet in red is an iron-deficient diet, and green is an iron-replete diet, and then in blue is a iron-supplemented diet. And what you're looking at on the y-axis is how much iron there is in the feces, very simply. So the more iron you eat, the more iron is going to be in the feces. But what was a little more surprising is that if you give IV iron injections with iron dextran, or simply 6 RBC unit transfusions over 6 weeks, you can also, these are mice that were on the iron deficient diet, you can increase the amount of iron in the feces simply by giving systemic iron. With mice on iron replete diet, the same trend happens. So with IV iron or red cell transfusions, you can increase fecal iron. But with the mice that were on a supplemented diet, there's so much iron already in the feces that we didn't observe any difference in fecal iron. And so then we simply looked at the feces, the microbiota that exist in the stool. And this is what I'm showing you here is a principal component analysis of the beta diversity, or the diversity of the microbiome in these mice. And what you can see is, if you separate it by diet, you could clearly see that the group that was iron deficient clustered separately from the group that was on an iron replete diet and an iron supplemented diet. When we look at the mice on an iron deficient diet, the mice that were given IV iron or transfusions also the ones that were given systemic iron clustered separately from the mice that were not. And so by giving the organism iron systemically, you can affect the gut microbiota. Same thing happens for mice on an iron replete diet. On an iron supplemented diet, it's less clear. So to make a long story short, whether you use the fecal iron concentration, the organism's ferritin level, or the hepcidin level in the organism, there's a good correlation with certain bacterial taxa. For example, lactobacillus, and this is well known from other studies as well, the more iron you have, the lower the lactobacilli levels, and that's thought to be one of the protective bacteria that's in your gut. So at the end of the day, why does this matter and why are we studying it? And so the underlying clinical problem is that patients getting stem cell transplants are often iron overloaded from the need for red cell transfusions. And it turns out in epidemiological studies that increased transfusional iron overload is a predictor for increased mortality. Now, these human studies have a confounder by indication in that the patients who have more iron were given more transfusions, so may be more sick to begin with. And so it's a little unclear whether this is due to the iron load versus the need for the transfusions. But nonetheless, there is epidemiological data to suggest that increased iron leads to worse outcome in stem cell transplant patients. And so again, we model this in mice. We take mice that, and we either keep them as controls or give them IV iron. And then we can give them chemotherapy and conditioning regimen and give them a stem cell transplant. And in this case, we're giving them green bone marrow so that we can track them more easily. And to make a long story short, if we look 28 days after transplant, the mice that were iron overloaded, first of all, you can notice that there are fewer dots here. They didn't start with fewer dots. It's just that they don't survive as well as the mice that weren't iron overloaded. But also, if you look at the percent of GFP cells in the circulation or the hematopoietic recovery, it's much lower in the mice that were iron overloaded. And so because there are a number of studies now linking the microbiome to stem cell transplant outcomes, we're very interested in seeing whether the iron load caused by the red cell transfusions by impacting the microbiome can also affect stem cell transplant outcomes. And those are experiments that are currently underway in the lab. And so finally, I want to transition to what happens when you have too little iron and iron deficiency in blood donors. So hopefully I've shown you a little bit that too much iron can be harmful, but what about too little iron? And so in the United States, at least, we need about 13 million blood donations per year to meet our supply demand. And only about 5 million Americans actually go out and donate blood. And so most of the donors are repeat or frequent blood donors who are donating 2 or 3 units on average per year. And we know from the epidemiological studies that when you do this, up to about 50% of women and 30% of men do become iron deficient from these frequent donations. And is this the problem? I would argue we still don't know. We came up with the hypothesis more because we're very interested in storage quality is, might iron deficiency explain a cause of poor storage quality? And so again, we first did these studies in mice, and on the y-axis, you're looking at the post-transfusion recovery, which is the gold standard marker for the quality of blood. And and these are mouse studies, so if the mice that donate blood are iron replete, we get about a 75% post-transfusion recovery, which is the sort of the FDA standard. But as you decrease the amount of iron in the donor mice down to IDA, which stands for iron deficiency anemia, IDE is iron deficient erythropoiesis, you can see that the post-transfusion recovery has a stepwise progression, and it decreases with decreasing iron in the donors. And so there may be a situation where the less iron your donors have, the less well those red cells will circulate in the recipient. And so we wanted to test this in humans, and unfortunately, because of the pandemic, I can't show you any of the data. So the study was delayed by about a year, and so we won't complete it until the end of this summer. And so maybe next year I'll be able to present some results from this study. But what we've been working on for the past 4 or 5 years now is we've been recruiting healthy blood donors who are iron deficient from their blood donations. They donate a unit of blood, which is iron deficient. And then 42 days later, we do a post-transfusion recovery study with radioactive chromium to assess their red cell quality. On the next day, we randomized them to either IV saline or IV iron, and this is a full gram of iron dextran. And then we wait 4 or 5 months and then have them come back again, and they donate another unit of blood. If they got iron, we presume that it's going to be an iron replete unit, and if they don't, then they will remain iron deficient, and then we do another post-transfusion recovery study to be able to compare to see if it affects the quality of the blood. We also took this opportunity to do a number of neurocognitive tests, so we do functional MRIs and very sensitive neurocognitive tests to see whether iron deficiency in a blood donor may affect any aspect of neurocognition. We're also collecting fecal samples to examine the microbiome, and that's not shown here. But again, unfortunately, because of the pandemic, I can't share any of the results, but maybe next year. So in conclusion, red cell transfusions are all associated with hemolysis from storage, and whether that's a problem or not remains to be determined. Your iron status will affect the microbiome, and this may be clinically relevant in a bone marrow transplant setting and potentially other settings. And we currently have a study underway to determine whether iron deficiency affects the donors and the recipients, and hopefully we'll be able to share those results in the coming year. And these are my closest collaborators here at Columbia in New York, who I get to work with every day. And these are some of the external collaborators that I also have the pleasure to work with. And I thank you for your attention.
Thank you all. It was super interesting. Yunping, will you take over from now, or do we have time for questions?
I think we have time for question this subsection. Now.
So does
we already have some.
Yeah. Okay. Good. I can't see them. If anybody has questions, unmute yourself and ask them, please. I can ask a question to you, Eldad. It's very interesting that you have actually shown that iron deficient blood donors' blood store worse than in iron replete donors. So do you think it will be harmful to give them iron tablets?
Well, so what I showed is in animal models that iron deficiency affects blood storage quality. So we still don't know for sure in humans. There is some older data to suggest that red cells from anemic individuals don't circulate as well. But of course, we typically don't collect blood from anemic individuals. And so I think time will tell. Whether to give donors iron tablets or not is controversial, and it will depend on what the impact is, or how much of an impact there is on storage quality from iron deficiency. If it's tremendous, then I would argue either offering donors iron tablets, or increasing the duration of length between donations, or testing them for iron deficiency and not letting them donate if they're iron deficiency may be more optimal. Ultimately, I think what we need is more people to donate blood so that we're not dependent on so few people donating so that they become iron deficient. And so if everyone donated more, we wouldn't have as much of a problem.
Thank you. In Denmark, we have for some years now offered iron tablets, not to take every day, but in kind of an intermittent way. And we are down to very few who are actually iron deficient. Of course, we haven't tested their microbiome, but I think that works very well.
So we don't, I mean, it will impact their microbiome, whether that's harmful or not, and in which situations it might be harmful, we don't know.
I'm kind of hoping, given it in an intermittent way, it will reduce the impact of the microbiome. Yes, Stephen, you can. Did you want to say something? Okay. Okay. Any questions from anybody? Then Yongping, you can take over. Thank you.
Thank you. And welcome to this subsection on donor recruitment, with the focus on donor recruitment and the donor retention from the family and emotional donations to sustainable voluntary regular donations, lesson learned and the challenges. We will have three presenters. Dr. Andre Lua, who is WHO regional advisor for blood safety and also for tissue and transplantation. And we also have Mr. Gianfranco Massaro. Perhaps everyone know him, the president of FBDO. FBDO is one of the 4 founding organizations for the establishment of a World Blood Donor Day, together with WHO. And we also have Professor Michael Clement from Hamburg University. I don't know whether that's accurate or not. Actually, I just searched it in Wikipedia about Professor Michael Clement. But before that, I am supposed to give a very short presentation on blood donor recruitment. So, effective blood donor recruitment program is the foundation for sufficient and sustainable blood supplies. Information for this year's World Blood Donor Day, we mentioned the effort for people to donate the blood and the plasma during the time of this unprecedented crisis. And I said that this extraordinary experience demonstrated, perhaps I should say that once again, the crucial role of well-organized, committed, voluntary and unremunerated blood donors in ensuring a safe and sufficient supply during normal and emergency times. And apparently, it is also crucial for the successful implementation of the trials of convalescent plasma in treating COVID-19 patients. So come back to my first slide. At the left corner, you can see that this is the image of a 2 WHO publication, or one publication. Another is a company training tour on donor recruitment. And so this document actually includes the strategy for the donor recruitment. It's a joint publication for WHO and IFRC. Although today this is not a focus, but I want to say that given the importance for donor recruitment, WHO and in cooperation with IFRC in the past has organized a lot of global, regional, and national capacity building activities for donor recruitment. I remember even WHO once bring a whole team of experts or experienced donor recruiter from the world to visit one country and organize a training session, and actually mobilize the country to change from family or paid donation to voluntary donation. Anyway, this document is, the purpose of the document is to support the countries to develop a donor recruitment program. And the document is based on the program or the best practice of donor recruitment and retention in the world. So this slide shows all these strategies in this document was organized under the 4 strategic goals that identified and achieve to achieve an effective donor recruitment program in the countries. The first goal is the government commitment, societal support, and the setting up of the effective national blood donor program. The second is to foster a culture of voluntary blood donation through increased public awareness of the need of blood and community participation. The third goal is building and maintaining a safe blood to maintain a sustainable voluntary donor base through the recruitment and the retention program. The fourth one is to provide a donor with quality donor care and service. So here are the more detailed strategies or the programs to achieve the goals. The approach to increase public awareness and also the community participation might include understand the donors through surveys, develop communication strategies, proactive involvement, working with media, mobilize the community partners. These community partners could be a local Red Cross, Red Crescent Society, faith-based organization, schools, factories, or like in Italia, the national or the local blood donor associations, if it is available. And also maximize the impact for blood donor and the local and the international blood donor event, for example, or Blood Donor Day. The strategies and the programs for the recruitment and the retention might include educate, motivate and recruit new blood donors, mobilize the youth as a new generation of blood donors, convert family replacement donors to voluntary and repeat blood donors, implement targeted retention programs, and the recognition for blood donors' contribution to healthcare system and also to society. For example, this is something usually happens in many countries to award the donors and also to show the recognition for the blood donors. And certainly they provide a quality care and make a blood donor a safe and a pleasant experience. This needs the competent and professional staff, standards and appropriate procedures. It is important really to make blood donation a safe and a pleasant experience, make it convenient for blood donor give blood, ensure meeting donors' customer experience, the expectations. And finally, but actually this is very important, especially for countries where there is no such a blood donor program, create an enabling environment and set up a voluntary non-remunerated blood donor program. So this need to have the government recognition for the importance of voluntary donations and also commit and support to voluntary non-remunerated donations. Establish a national voluntary blood donor program with a policy plan and legislative support, adequate resource, facility, equipment, and also train staff. And also have a broad collaboration, partnership, and networks from wider societies. So again, this is to this overall slide for all these strategies. I want to say that first, the objective goal is actually important to support the implementation of retention programs, because it is crucial that the donor donation process is safe and the donor have a pleasant, satisfied experience when they first donate blood. Another thing I want to say is that the donor recruitment strategy or the intervention is evolving. And one example is that in this WHO and IFRC document, the use of IT technology and information technology was mentioned, but just as a creative way to attract young donors. But today, as we know, that the use of internet IT technology, especially the smartphone for the donor recruitment, retention, and even to conduct the donor recruitment research, now it's become a very popular, very important strategies. And at the same time, there has been more and more both theoretical and programmatic studies on donor recruitment. Thank you. So I think now we can have Mr. Gianfranco Massaro for the first presentation.
Buon pomeriggio a tutti.
Good afternoon, ladies and gentlemen. My contribution will entail the retention of donors, so the experiences carried out and the emotions involved. But before doing that, I'd like to briefly introduce the FIODS, which was established in Luxembourg. I think someone is sharing my presentation. This is our organization. We have the General Assembly and we have an executive committee, continental delegates, and the Solidarity Foundation, as well as a steering committee and the international committee involving young people. So these are our four main partners. That is to say, they represent our world level of excellence. These are the European countries which are part of FIODS. We also have Canada and the United States. We have 14 countries in Latin America, and that was an extraordinary success. Then we also have 10 countries in Asia, which are involved in our association, and 15 African countries involved, in which are member of FIODS. So FIODS is also registered in the Economic and Social Affairs of the United Nations, and also is part of the Conference of the Council of Europe and the Conference of NGOs of the Council of Europe. So we gather 33 million blood units at world level. This is an important information to convey to all of you. And we have 27 million blood donors. Furthermore, we have 1 million donors which are paid, which are remunerated for such activity. Next slide. And here I like to share with you some of the projects we launched when it comes to the retention of the donors we have. It is not easy to have a volunteers association which is based on a free of charge solidarity and social commitment, and cultural, regulatory, social, and let's say habits differences may make it difficult or impossible to for instance, shift ideals and operational procedures from one country to another. And in Bolivia, for instance, we developed a project, Dar Sangre es Dar Vida, that so, for instance, Fiats and Aves, and we established the Association of Bolivar of Blood Donors. So this is an important initiative, that is to say, an initiative of ABDS, that is to say, the promoter, a lady which welcomes the donor and helps the donor to fill out the questionnaire as well as the other documents which are needed before blood donation. So this, of course, entails a close collaboration with the public facility of the Banco de Sangre, which has to recognize and acknowledge this, let's say, activity. Furthermore, it was decided to clearly identify the blood donor by asking this person to wear a red jacket, which identifies that person as a volunteer. And therefore, preferential access is agreed to donation. So these are little things, of course, which may, however, lead to a retention. And this idea of the association led to an increase of donors that register in the association, which become regular donors. And therefore, it is important to have volunteers which are suitably trained and in the places of encounter of young people like schools, universities, and gyms. So this was very much fruitful in Bolivia, especially when it comes to the presence of volunteers that talk about their experience as donors in universities. And they are accompanied by healthcare providers that, for instance, determine for instance, the specific blood group of the people that are there. Furthermore, in 2018-2019, there was an extraordinary project financed and funded by the Italian government worth 600,000 euros, where Fiats and and Aves of Italy were involved. And therefore, we worked on 2 Latin American countries, that is to say also Salvador and Guatemala. In these countries, actually, we had no volunteers associations, and therefore we established those associations, and in fact we trained actually the donors, we trained the people that could actually play a role in the association. Then we also had a period of training and growth, both on the side of volunteers, which came to Italy, and also together with the doctors and the nurses and the experts that visited a lot of Italian facilities, and then they realized what the situation is in our country. And of course, our Italian model cannot be exported to developing countries. But generally speaking, we organized a number of stages, which were very important. And then we succeeded in training 2 associations, national associations, and also local associations. And hence, with the support of Aves and Fields, and we also gave to them and donated to them some materials, like, for instance, portable fridges and welders and anything needed in order to, for instance, organize the various centers. I wish to say that in some of the countries I visited in Latin America or in Africa, with a portable fridge, which costs 200 euros, 300 euros maximum, it is possible to increase in those countries by 10% the level of blood donation. Because, well, although strange it may appear, it is difficult to take blood from one place to the blood center. So, of course, this makes it possible. And then in Guatemala and in Salvador, we have organized some facilities and organizations. And another important project in Burkina Faso led us to train some partners. So we organized 4 theatrical shows in the capital city. And also, we organized a stand at a cinema festival, and we organized, for instance, a tour organized by Blood for Africa Foundation as a part of the recruitment of blood donors, and also participation in a number of workshops from the 13th to the 16th of February in Tendogo on the fight against HIV/AIDS. So over the time, this campaign was fruitful and increased from 2015 to 2018 by 16,000 blood units. Of course, the goal has not been achieved yet, but the progress is quite satisfactory at the moment. And recruitment and also selection and retention of donors are among the main concerns that we have. And in fact, in consideration of the young age, and we know that in Africa we have a lot of young people, so continuously recruiting donors without a retention policy will be like starting from scratch.
And this mission is very difficult to obtain or to achieve. Very few donors have an address through which they can be contacted if needed. Less than 5% of the population has a telephone or a post address. There are shortcomings in means of transportation, and then, of course, there is the high cost of life. These are the main problems we encountered in Burkina Faso. And considering the current state of things in Burkina Faso, our proposals to improve the situation is the following: the promotion of blood donations, the recruitment of donors, and the campaign brought about by the national campaign to promote donating blood is very effective, but it can still be improved in terms of training partners in donating blood and in the communication with the media. In 2015, training was also extended to other partners, especially those who teach sciences of life and earth sciences in high schools, who are identified as being capable of actively participating in disseminating information and in educating towards donating blood. Training and education was offered to all teachers in high schools and institutes, and it cannot only be limited to those who teach life sciences and earth sciences as it used to be. 58% of the blood bags collected in 2018 stemmed from mobile collection. And the current context, with a very high cost of living, an increase in the price of oil, and shortcomings in public transportation, means that blood donors, despite their goodwill, will become increasingly posed by obstacles in reaching these donation centers, and therefore the alternative would be to strengthen mobile collection. We had some important information campaigns. You can see an image of our campaign at the National Police School. This is the National Identification Office. They organized the National Identity Day. We have a picture of the Director General of the Institute. So this is what we worked on in Burkina Faso, and we have seen a little bit of progress. Now I'll be showing you some images I collected during my travels. These are images from a donation in Senegal. As far as Senegal is concerned, now to Westerners, of course, take a look at the state of things when it comes to donating blood. And those of us who come from Europe, from more advanced countries, we have to help these countries. Next slide, please. This is a picture taken in India. Next slide, please. Nepal. Next, please. Guinea. In Afghanistan. Thank you. Thank you very much for your attention.
Thank you very much, Franco. And thank you very much for last year sending equipment to Nepal and Ghana. The service in these two countries. Very good suggestion. Gesture to people really appreciate. So next presenter is Dr. Andre Noah. Andre, please.
Hello. Did you see my screen?
Yes. Yes. Yes, we can see you and your camera too. Please.
I have. It's okay now.
Yes.
Okay. Thank you so much. I would like to thank the organizing committee of this symposium for giving me the opportunity to present the experience from the WHO Afro region on donor recruitment and retention. The outline of my presentation will be the following. I will start by a brief overview on the WHO Africa region. The region covers currently 47 countries with over 1 billion inhabitants, which is about 14% of the world population. We have 3 sub-regions to facilitate the WHO work in the region. We have Central Africa in green, Eastern and Southern Africa in purple, and West Africa in red. The need of blood transfusion in the region is very important because of heavy burden of infection disease such as HIV, malaria, tuberculosis, but also a high infant and maternal mortality, as well as mortality due to non-communicable diseases and road traffic. We have also a high prevalence of anemia. We all know that the availability of blood and blood products are global concern, and the WHO has adopted several resolutions through the World Health Assembly and the regional committee, urging member states to develop national blood transfusion services based on voluntary, non-remunerated blood donation. This slide recalls the main resolution at the global level, but also at the regional level for access to safe blood and blood products. Recently also, my colleague from headquarters talked about this. A framework for access to safe blood product was developed, and there are also other guidelines for access to safe blood and blood products. In 2021, the regional office for Africa adopt a strategy for blood safety in the African region. One of the objectives and targets for the voluntary non-remunerated blood donation was to assist countries in the region to set up an effective system of recruitment of low-risk donors, and at least 80% of all donations in countries in the region should come from voluntary and non-remunerated blood donors. What are the strategies used in the region for the recruitment and the retention of voluntary blood donors? There are many, but I only summarize those are commonly used in the country. I take this opportunity to thank all the countries that have provided me information needed to prepare this presentation. Among these strategies used, there are the following: public awareness campaign through advertising campaign and use of conventional and social media like radio, TV, newspaper, Facebook, Twitter, Instagram, etc. for inviting blood donors for the subsequent donation, thank the donors, and the remainder, and even notification. But also, other strategy is to engage partnership and collaboration with government agencies, schools, and tertiary institutions, media, corporate, and faith-based and non-governmental organizations, as well as with rural and urban community. In some countries also, they create youth club 25 or 30 and other peer education and promotion program for recruitment of blood donors. Other strategies is to celebrate national, regional, the World Blood Donor Day, that are important events in the region for the recruitment and the retention of blood donors. During these events and other occasions, blood donors recognition and awards such as gifts, medals, certificates, shield, other incentives are important for donors' retention and to encourage people to donate blood. There is also a strategy to advocate with national authorities for funding and support blood donation activity. During the COVID-19 pandemic, in addition to public awareness campaigns that are already implemented, some countries put in place transporting donors from and their home with authorization from national authorities using all the means that can transport donors, but also through establishment of call center where some way to recruit and retain volunteer donors during the lockdown to reach out blood donors. What are the main outcomes? For the availability of blood and blood products, this graph shows figures of whole blood donation. The total number rose from almost 2,003,000 blood units in 2002 to around 5 million blood units in 2018. The average blood donation rate per population were almost 5 per 1,000 population, and 8 countries in green on the map were collecting at least 10 blood units per 1,000 population or more, as recommended by WHO. The map shows also the blood donation rate per 1,000 population per each country in the region. The trend of the types of blood donation on this graph show an average percentage of voluntary blood donation that was 56% in 2002 and 71% in 2018. In the region, some countries reported also pay donation. In 2018, 19 countries in green on the map in the region have achieved the targets of the regional strategy for blood safety. That is, I recall, at least 80% of blood donation from voluntary, non-remunerated donation. With regard to the impact of the COVID-19 pandemic on blood supply in the region, the progress on access to blood could be lost because of this pandemic. Indeed, we conducted a quick survey on the impact of the COVID-19 pandemic on blood supply and demand. The results were published in Vox Sanguinis and are accessible at the link indicated on the slide. Overall, blood donation, blood drive, blood demand, and blood issue and transfuse dropped in the region with an average percentage in a bracket in the blue color. What are the main challenges in the region? In the region, we have ineffective coordination of national blood services in most countries. Also, infrastructure, financial and human resources for the recruitment of voluntary blood donors are inadequate. We have also a low public awareness arising from deep cultural myths and misconception, but also from religious reasons. Some countries are applying cost recovery system for blood product through out-of-pocket payment in most countries. This creates a lack of trust and motivation among voluntary blood donors. In our region, as you have seen, there is a persistence of family and pay donors through orphan hospital blood banks. We have also in the region a high prevalence of transfusion transmissible infection in the population. Furthermore, emerging and re-emerging infection of public health importance, such as COVID-19 pandemic, is impede the voluntary blood donation. Some lessons learned. Partnership and good collaboration between different stakeholders are vital for blood donor recruitment. Effective public awareness and information is also crucial to reach potential donors. Blood donor recognition events are necessary in increasing the needed awareness about voluntary blood donation. Sustainable financial resources from government and skilled personnel are needed to achieve 100% of voluntary blood donation and promote repeat donation. The COVID-19 pandemic negatively impacted blood supply due to limited access to blood donors in most countries due to lockdown, donor anxiety or fear of COVID-19 infection during blood donation, which often stem from popular misconception and misinformation that have hindered blood donors from accessing blood transfusion services. In conclusion, blood and blood products play an important role in healthcare, in particular for vulnerable people such as mothers and children. The availability and timely access to safe blood and their optimal use are essential for health system strengthening and for the achievement of universal health coverage. Many thanks for your attention.
Thank you, Andre. Thank you very much. We continue to invite the next speaker, Professor Clement, to give a presentation. Professor Clement is from the Institute for Marketing of the University of Hamburg.
Well, thank you very much. I hope you can all hear me.
Perfectly.
Perfect. Well, thank you very much for the invitation. My name is Michel Clement. I'm a professor for marketing at the University of Hamburg, and I have the honor to present you some insights that have a very large title, namely the European region, marketing decisions and blood donation. Now, I know that between you and your well-deserved glass of white wine for the evening is just my presentation, as I am the last one in this road. So I will try to make it short and precise.
Great.
I actually work together with the German Red Cross. We have installed at the University of Hamburg a research center that addresses marketing questions with respect to finding insights, how to incentivize, how to retain, and how to regain blood donors. And of course, as I am from the marketing department, I have this claim that marketing is a very important management task to actually win, retain, and reactivate donors. And what I've pointed down here in this slide is that we are interested in the beginning of a donor life cycle, and also we are interested about the end of the donor life cycle, which is not the natural death, but the decision by the donor to not donate anymore. Now, what we do is, and that's something that I would like to pin down and would like also to motivate all of you, is that we run together with the German Red Cross, but also in projects that we run together with the Austrian Red Cross or St. Queen in the Netherlands, that we actually run experimental tasks to actually test the marketing impact on donation livelihoods. So we have predefined settings where we are actually trying to integrate a manipulation and then see whether or not we can activate first-time donors or reactivate former donors. We also believe heavily in data science, which requires that, at least from the marketing perspective, that you actually collect the data that is related to the communication to each of your donors. So ideally, we can match marketing instruments and marketing activities by your organization towards the donation behavior of your population. So given that we can do this, we can then work together in the optimization of the marketing mix, which actually means when do we send what kind of incentive, what kind of letter, what kind of message to what kind of donor. So a segmentation and optimization of marketing instruments with respect to the segments is key, and I will show you some of our research results in this field. We also strongly suggest some changes in the referral management because we know that referrals are actually hurting the likelihood that somebody is coming back to donate blood. But we also propose a few sets of strategies to overcome the pain, at least a little bit to overcome this pain. Last but not least, we also ran experiments to test reactivation strategies. And I would like to show you now just a few insights that we have gained from the projects. And I'm not going to go into the very details of each of these findings, but I would like to come directly to our findings so that we can then, if you are interested in, contact me directly to discuss further issues. So what have we done? We did, for example, study the impact of incentives on donation. And we have, and actually I would like to actually provide two main implications. First of all, we have shown in the paper that has been published in the marketing field that health checks are a very powerful incentive. We ran two large-scale experiments in Germany with the German Red Cross in Baden-Württemberg and Hessen, and also in Baden-Württemberg, where we had a control group and a treatment group. And the treatment group received a health check for the third donation. And we have large treatment groups and we have large control groups, and we see that the invitation for the control group, the response rate, that means you receive an invitation, and the likelihood that you will then donate blood was 38.4%. So we observed the response rate of 38.4%. And you see that the treatment group, that's a group with the health check, had a much higher response rate of 44.9, which is the absolute difference of 6.5 or the relative of 16.9%. This is a powerful impact. We also reran the experiment to also observe whether or not there are some effects that wear out over time. So maybe a health check is only interesting for those that do it for the first time, but we don't see actually this negative effect. We see again when we reran the experiment at a different point of time and some kind of a different area, we see again that we have an absolute increase of 3.1%, which is a relative increase of 8.2%, just by providing a health check for those that are going to donate for the third time. So my point is, if you are actually thinking of providing incentives, a health check is a real powerful instrument. Second, we are currently working on testing strategies which are related to incentives. And what we do find is that if you provide donors not with money, but an incentive like a little present, which is unexpected and which is an hedonic, that works much better than providing them with a utilitarian incentive. Now, there are all kinds of different psychological mechanisms behind this, but the key managerial impact is if you want to provide an incentive, think about a health check, or if you want to give a present, take an hedonic present. We also looked at what kind of motives are triggered by campaigns, also when you are related to negative impacts or negative events such as a pandemic or a catastrophe. The most powerful instrument in this case is actually working with the public relation to ensure that outsiders provide the information that you do need blood. It is better that everybody else talks about this topic than if you only talk about this by yourself. We also have been working on donor referral programs, but I don't want to go into the details here. Now, we also then, if we are looking at the impact of retaining donors, we know that if you send them an SMS with respect to thank you very much for your donation, your blood has been used and at this point of time in a hospital, that it is best to send out this SMS directly when the blood has been used, so that the donor gets more or less right away feedback once the blood is being used, and not as a marketing tool at the very end to, let's say, reactivate the donor to say, well, you are eligible to donate in a few weeks, and remember your blood was used this and that time. So the timing of the SMS is a crucial topic, and it is best to send out the SMS when you say, thank you very much, your blood has been used at this point of time at a certain hospital, when the blood is actually being used. We also know that deferral strategies are very important. In a just recent paper that we have been published in Transfusion, we looked with using data from the Austrian Red Cross, how negative deferral is and how much this is dependent on the experience of the donor. So the assumption is that if you have more experience as a donor, the deferral is less harmful. And this is actually true. We see that the more experienced a donor is, the less harmful a donation is. So you see this upward trend. But the ugly story of this upward trend is that it's still a negative one. So that means if you defer somebody, we always observe a negative impact on donation likelihood. That's not really independent of the experience. The more experienced this donor is, the less harmful it is, but it's still a negative effect. However, the effect is very strong if this person is being deferred for the first time. So you need to implement, at least from our point of view, strategies to overcome the negative impact of a deferral, especially for donors that are being deferred in the very early stage of the life cycle. Now, what are these potential strategies? We would propose two strategies. One is you should give them a new date when to come back. So if you have to defer somebody, give them a date that you can hopefully plan to ensure that this person is going to return. At least this works in the German market very well, because when in Germany you hand out somebody an appointment, they stick to it. Second, you can also think of strategies that you send somebody home and this person has not achieved its goal or his or her goal, which means that there is still this open motivation to do something good. So what we also figured out in experiments is provide them the opportunity to make an alternative good deed if you have to defer them, which could be, for example, writing a postcard to an elderly house. Last, I think it is very important that you think about reactivating your donors. What we did is we tested a reactivation strategy together with the German Red Cross. And what we did is we sent donors that were inactive, and we defined inactive as being not donating blood for more than 2 years. You can send them your normal standard letter that blood is needed and you are asking them to return. Or what you can do is you send them a letter where you explicitly state your blood was used 2 years ago in a certain hospital and you saved somebody's life. And the likelihood that you are reactivating this person is being increased by about 11%. So if you are actually planning to send out reactivation campaigns, our suggestion would be to tell them where their blood was used the last time when they were donating blood. This at least is much more helpful in reactivating donors than not doing this. I would like to stop my short overview with just an announcement that we are hosting the 4th European Conference on Donor Health and Management together with the German Red Cross in September 15th to 17th. So if you're interested to join, then please visit the website ecdhm.org or look at the website of the EBA, where this information is provided as well. That's all from my side. Thank you very much. Thank you very much for the invitation. If you have any questions, I'll be happy to take these either via mail or later in the Q&A session. Thank you very much.
Thank you very much, Professor Clement, and thank you very much for taking interest in this this area. I know you're a professor of business administration and in a very distinguished Hamburg University. So I think it's very important, really, to conduct this study or the evaluation of. Basically, the donor recruitment and retention is not regarded as a solid science, as a clinical procedure. So there's a need really to regularly, to study and update the strategy. I think we already have some questions. For you, Professor Clement, or ask
yes,
to comment?
I can pick up. Yes, I can see them. So first of all, the risk of incentives. Yes, there is a strong risk of incentives on the one hand. And of course, I know the literature of these crowding out effects, but I'm not talking about putting down money on the table. Putting down money on the table is a useful strategy. It works. At least different papers have shown these, that there is a substantial impact on this. But I completely agree that this is not the path we should proceed on. But nevertheless, we have an exchange relation with a donor. And when I say incentive, I don't necessarily mean that you need to give them a bottle of Chianti Classico for 25 euros. That would be no doubt an hedonic present. But giving them something which is not really expensive, but it is somewhat of a hedonic experience. It could be chocolate, but don't give them a utilitarian incentive. But nevertheless, I believe that incentives are a powerful tool. And if you need donors and you have to retain donors, and if you have to incentivize donors due to whatever reason, I think a health check is something that is absolutely in line with the goals of the WHO. A health check is something in line of the health of the donor, and it helps you as well as the donor to actually provide this information. And what we at least see in the German market, that really works. So based on 2 large field experiments, I'm a big fan of these health checks. The second question I can see in the chat is it possible to use social marketing? Yes, of course it is possible and necessary to use social marketing. I mean, if you look at the marketing, the general marketing topic. So if you want to address the masses, forget about doing the advertising only on television. The masses are not on television anymore. A few parts of the masses are on television. You can reach them on television, but most of them are so decentralized so that you can actually only address these by social marketing tools. So you have to go, or otherwise you have to fish where the fishes are. And if the fishes are on social media, then of course you need to spread the messages via social marketing.
May I add just a comment? When you talk about the health check to the donor, I'm not convinced at all that this is an incentive. This is something that we need to do in order to protect in some way the health of the donor. Remember, we discussed in the previous session about the danger of depleting iron in donors, and there is a very nice editorial in Blood, I think, whose title is "The Price of Blood is in Iron." So whenever you look at the metabolism of iron in our donors, they are doing something that is very important for protecting the health of donors. It's not an incentive. It is something that we do for protecting the health of our donors. And if the result of the studies presented in the previous session are so exciting, that means also that probably we are also protecting the health of the blood that we are collecting. So sometimes I think that, and I believe that doing something that is not considered normally in the follow up with the donors, and in this kind, an health check is not more an incentive to the donor than something that is good for us, for blood establishment, to protect the health of the donor, and possibly to protect also the health of the product that we are collecting.
May I answer this? Of course, I would definitely agree that the donor health is the most important part of the whole thing. I never actually doubted this. My only point is, you do test anyway. And if you give the donor feedback on the status of his or her health parameters, that is something that you provide them back as an information. And that could be, and that should be, an incentive. You can frame it as an incentive in a way that you take a little care about your donor base. Of course, they know you will call them when there is a real trouble issue. But just to give them an information, they are healthy, and that those are the parameters, is, at least from my point of view, a nice incentive, and it is directly related to actually ensuring donor health as well. So it is a matter of framing this wellness behavior in a way, and it's an option to at least retain donors that are health conscious.
A win-win strategy.
Exactly.
I have a question to Franco. When the AfriPDO provides support to countries in Africa or in other regions, do you do that through the local blood donor associations, or you do that through the National Blood Service, or the National Blood Donor Recruiters or blood donor managers, where usually they're based in the blood service?
Certamente.
Well, both things, actually. We get in touch with the associations directly, but we also have a dialogue with institutions, of course, because our role is that of, in fact, dealing with institutions. We never bypass them, going directly to donors. So both things are important, and you yourself know that when the IFBDO gave some scales, I can't remember exactly to which African country we went through the WHO, and they asked you to see which African or Asian countries had this need for scales so that we could make the donation. So we talk to the institutions, but we also speak to donors, of course.
Yep. Do we have more questions?
I shouldn't have said that with the white wine.
We have the bottle under the table.
So if not, I think now I give the floor to Dr. De Angelis to close the symposium.
Thank you very much. Thank you very much indeed to all the presenters, to all the speakers, and we are going to close this today's scientific symposium held in the framework of this 2021 World Blood Donor Day. And of course, I would like to thank all the presenters and the chairpersons who guided all the symposium and did their best to give us a deeper insight of international and country perspectives and regional perspectives throughout the world on horizon scanning, and mainly dealing with convalescent plasma and immunoglobulins, on contingency plan to ensure an adequate blood supply, and on donors management, from donors health protection to the best gift as a transfusion product. And what I've observed, in my opinion, the discussion has been very helpful. And I would like to recall that the discussion gave us the opportunity to share enthusiasm on possible collaborations between institutions, professionals, scientific bodies, and donor associations for the improvement of a transfusion system based on voluntary, non-remunerated blood donors and donations, leading to the sufficient availability of safe blood and blood products, including plasma-derived medicinal products. The end of this symposium is also closing the celebration for this 2021 World Blood Donor Day. And this annual global event aims to pay tribute to all the voluntary donors for their valuable gift needed to secure high-quality blood for its use in the healthcare system throughout the world. In 2020, COVID-19 pandemic dramatically broke in, and the possible exposure of blood donors to a potential deadly airborne infection during the donation process is something unprecedented in my experience as a transfusion medicine doctor. And nevertheless, throughout the COVID-19 pandemic, despite limited mobility and other challenges, blood donors have continued to donate blood and plasma to patients who need transfusions. This extraordinary effort during this time of crisis highlights the crucial role of well-organized, committed, voluntary, non-remunerated blood donors in ensuring a safe and sufficient blood supply during normal as well as emergency times. It has been a great honor and a great responsibility for Italy and for our organization and for the National Blood Center to host this first virtual edition of the World Blood Donor Day after 2 years' silence. It is clear that the main weakness of this edition are the limited physical presence, the less chances of attracting passing by citizens, and the different troubles in the organization, with many changes in place during the preparation. However, we would emphasize some big opportunities represented by the wide impact in a global digital framework and the different target groups that have been addressed at the same time by different means. And in conclusion, we are confident that the pandemic challenge became an opportunity to demonstrate the resilience of the mankind, not being overwhelmed by difficulties. And thanks again to everybody. Give blood and keep the world beating.